I think we'll go ahead and get started, everybody. All right, so we're all excited to be back in the CNRA headquarters for day 2. A little bit of logistics. The agenda is available at the front desk at registration and also on the QR codes that are around. The bathrooms are underneath the stairs and to the right. Wi-Fi here in the building is CNRA guest. As a reminder, there's no food and drink in the auditorium, and we have to enforce that. So please abide by those rules. But when you're out in the lobby, you can have coffee. Thank you to our sponsors for the coffee, ICF. If anybody needs an accommodation, see Christine Job, the front desk, at the registration desk. As a reminder, the workshop is hybrid, so if you're online, you can submit questions through the Q&A Zoom. And then Sacramento Pipe Works is the after hours location, 4:30 to 8:30 for climbing if anybody wants to, or there is a bar in the same building. So with that, I'll hand it over to Rosie.
Thanks, Adam. All right, so for today's plenary session, I think we wanted to really do something to let all you know how important your research and monitoring is. So everyone here, most of you are the ones out in the field driving the boats, in the lab counting zooplankton, pipetting samples, filtering water, running qPCRs, entering the data, graphing the data, maybe writing some reports. And you're probably aware of some of the regulatory mandates that kind of inform your work or require your work. You know some of the management implications. You may have written grants that say, oh, if I do this, this will inform management because X, Y, Z. And hopefully, Kerrigan's talk yesterday gave you a little more of that context of how your work fits into the greater whole. But I know a lot of the scientists who are out on the boats every day don't always see the process of how their data gets into those regulatory or management decisions. Where do these new requirements come from? Where do the new permits come from? How does your data get into decisions? So today we have some scientist agency managers to talk about how their agencies have used IEP data to really inform new management decisions and basically enforce the environmental laws that we learned about yesterday. Now, this regulatory panel is maybe a little sparser than one would expect from something called panel. And to address the elephants that are not in the room, we were going to have some of our federal partners also on this panel, but due to the current political climate, they unfortunately had to bow out. Originally, this was going to be a much more interactive panel as well, but again, due to the current political climate, we decided that we're not going to be having a Q&A session with the audience, and we have a shorter panel, so we'll probably end up having a nice long coffee break, and perhaps you can continue the conversation over coffee in a less formal environment. So no question and answer period, but we will have Sam and Brooke talk, and we're also not recording this session just to make sure that people can speak freely. And with that, I will introduce our panel members, Brooke Jacobs from the California Department of Water Resources and Sam Bashevkin from the State Water Resources Control Board. Do you each want to say just a few words about yourself and then we can go into the talks?
Sure. Hi, everyone. I'm Sam Bashevkin. I work in the Division of Water Rights at the State Water Resources Control Board. And I'll talk about it more in my presentation, but I work on efforts to
Ahead and get, oh, it is on. All right, everybody, we're gonna go ahead and get started. Our next, we have session 7 next, flow and physical processes. Sam?
Hello, everyone. We're going to start the next session now. So I am Sam Bashevkin again with the State Water Board, and I'm here to introduce session 7, flow and physical processes. And let's see, the first speaker will be Jason Hassrick from ICF.
All right, thanks everybody. Exciting to be here with you. I'm going to be telling a story about a project that we just started just last year to test a conceptual model about how longfin smelt respond to changes in outflow. And the reason we're getting support to study this question is to identify the temporal window that is most beneficial to manage outflow and perhaps the magnitude of those flows that might be needed. So this is a large study, and I'm really just the talking head right now. I've listed my fellow co-conspirators here. Let me see if I can get this pointer to go here. I want to point out Wim Kimmer. He's the principal investigator. He's here in the audience. So if there's anything that I mess up, please clarify. Or if you have any questions that I can't answer, Wim's here as well. So, and then there's some other folks here, Andrew Kalmbach, Kim Brewitt, Ramona Zeno, who really did a lot of the analysis that I'll be presenting as well. All right, so here's the problem, right? Here, this is a model that Wim and Ed Gross published a couple of years ago, and essentially what you're looking at on the y-axis is the fall midwater trawl abundance index for longfin smelt, and you can see that over these years, the population of longfin smelt in the San Francisco estuary has been in decline, and pretty steadily in decline all the way until 2020 or so. And Wim and Ed published this model that really, so these yellow dots are the abundance indices, and then the model that was fit to that is this model that's listed down here. And essentially what it is a general linear model that has X2 and then a polynomial function for year in there. And you can see that it tracks pretty well until we get to like these most recent years, right? So what's happening here? And so that's part of what we're going to be trying to investigate with this study that we're embarking on. So, as I said, is the X2 standard that from January to June, is that really necessary? Is that something that could be potentially refined with better information from a better understanding of the mechanisms behind that relationship? And so this is probably a very familiar life cycle model for most of you in here, and what we're going to be focusing on is really this period where we're in the low salinity zone and we're transitioning into saltier water and what's happening with these fish. So part of the idea here is that they are using a sinking behavior to retain their position in the estuary, and there may be some beneficial components to that in terms of maybe taking advantage of more food resources that sink with them. There may be fewer predators, so predator exposure might be minimal at depth. And so part of this investigation, I think importantly, is to look at DL behavior, right? So what's happening at night, what's happening in the daytime, in terms of like what these fish are doing and how they're distributing in the water column. So what I'm going to do is play an animation that Ed Gross put together just to kind of give you an idea of what happens when particles sink in this estuary. And so if I can play this, of course it's not playing. Take off the pointer. If I click on it now. Well, so if I'm on pointer mode and I hit click, oh, is it showing?
Yeah. And then you're little and you're looking for
Hit that?
anything. Yeah.
Oh, no, sorry.
No,
You click again.
this one?
No, the one that you just clicked. Let's see if there's a.
It worked perfectly for the practice. Anyway, what I was going to show while this gets going. What a bummer.
Is it on the next one?
No?
Right, no. Yeah, because you had a button right there, right?
Yeah, there's a button.
I'm sorry.
Okay, well, I'll just move on. What I was going to show. What's that? Oh, there we go. Uh-oh. Now I really don't know what I did. There's the arrow. Okay, so now I've run out of time. If you drop particles at the Golden Gate Bridge and they sink, look at what happens. I mean, I think, intuitively we think that rivers flow into the ocean, and so generally flow goes into the ocean. But if these particles sink, this phenomenon called gravitational circulation actually brings these particles from salt water into fresh water, and you can see these particles that then migrate. As they go from yellow to green to blue, they migrate into fresher and fresher water. So the idea here is that organisms that take advantage of this gravitational circulation, or some people call it estuarine circulation, by sinking, can potentially retain their position in the estuary. And if their food also sinks, they could potentially take advantage of that and avoid predators perhaps that might not be going to lower in the water column as well. So anyway, thanks for bearing with me with that. I will try to catch up on time. And so one of the things that that Wim presented at the Bay Delta Science Conference was essentially like, so we've got if you model historical conditions, like in historical X2 conditions, right, or some kind of enhanced flow condition, so that maybe you put X2 where this green line is at 50 kilometers under higher flow scenarios, what happens to particles that either float passively or sink, right? So and what you can see here is, like, if you look at the green line and you look at the historical line, you'll see that particles that tend to float will tend to be moved into saltier water much more quickly under the enhanced scenario than under historical conditions. But if they sink, you can see here that these particles will actually be retained in the estuary more quickly and stay longer in the water column than under the historical X2 conditions. So under enhanced flow scenarios, and if you sink, you can actually retain your position better with this gravitational circulation. And so that's what we observe with longfin smelt. So now we're looking at field data, and you can see predominantly that these fish are spending their time in low salinity water. So here's a salinity of 5, and they're basically spending most of their time, up to about a salinity of 5. And if we look at this in terms of like their life stages, right? So now we've got these distributions of, so we've got bottom salinity on the x-axis here, and then we've got these different size bins of longfin smelt, from the really small ones, larvae from like 5 to 15 millimeters, all the way up to 45 millimeters. You can see that there's this tendency for them to try to stay in low salinity water as they get older and older, until they basically develop swimming behavior that enables them to then move into saltier water. So that's what that looks like. And then, so what might be a potential benefit for exhibiting this behavior? One idea is that there's just more food down there, like I said. And so what we're looking at here is one of their preferred prey items, Eurythema carolea, and these are some papers that have established that is one of their preferred prey. You can see that Eurythema carolea have been essentially declining in abundance in the estuary, and that is often thought to be in response to clams, right? So clams are either filtering out the phytoplankton, or they're potentially predating on the nauplii as well, and that's some of the work that Wim showed a couple of decades ago. And then, of course, how are these copepods responding to outflow? And we can see here that, under under lower X2 or higher outflow conditions. These are all different years here that are shown on this regression line. That food tends to be, or that these copepods tend to be more abundant under higher outflow conditions. Okay, so now we're going to move into kind of the field study that we've embarked on last year. And so, as I said, this is a daytime and nighttime study. We were using the same 20 millimeter net that CDFW uses. We also started out using the Smelt larval survey net as well in March. And so we were doing daytime and nighttime sampling in the low salinity zone, going out into saltier water than I think the monitoring surveys have done in the past. So we were starting at near bottom salinities around 20 and then moving eastward until we got into near bottom salinities around 5. And so here's what essentially those look like. We had a little bit of a rocky start at the very beginning, just getting our gear to work properly, but we eventually converged on essentially doing like 4 deep toes and 4 shallow toes per event, and then day and night. And this is the distribution of those sampling. So the white circles represent daytime sampling. The open circles represent nighttime sampling, and you can kind of see how, and the size of those circles represents the relative raw catch abundance that we had for Longfin smelt during this study period. So starting in March, we were starting out much more into San Pablo Bay and then moving into Carquinez Strait, basically ending just a little bit past Carquinez Strait. And then as flows kind of decreased over the season, that salinity gradient that set our sampling bounds changed, right? So this is not a geographically based survey, it's a salinity based survey. I think that's an important point to make here. And so this is one of the figures that WIM recently made based on last year's data. And just to kind of orient you a little bit here, so we've got these paired trawls, and whenever we went out, we would do a surface trawl, and then we would put a bunch of weight on the frame, and we would drop that 20 millimeter net down, and then do a deep water trawl, not an oblique trawl, but a deep water trawl. It ended up probably being a little bit oblique as we pulled it up, but anyways, what we're trying to show here is that those surface trawls and deep water trawls were essentially they were sequential, and so what you can see here is that for the most part, most of the larger abundance of Longfin smelt that we encountered were in the deep trawls. And that's pretty much the case throughout the entire survey period. And this is much more evident in the daytime than it was in the nighttime. So the fish are potentially changing their behavior a little bit more in the nighttime. And obviously this is just our very first year of data. So we're about to start our second year tomorrow. And so we'll see what this pattern looks like after this year. And so then the component of the study that I'm most involved with is actually looking at the potential predators for Longfin smelt. And these are northern anchovies. They're very abundant in saltier water in San Pablo Bay. And what we're doing is we're embarking on a study to try to quantify the relative abundance of northern anchovies that might be encountering these Longfin smelt in the low salinity water as they move into higher salinities. So this is just a plot of historical bay study data looking at anchovy catch as a function of salinities, right? So moving left to right, you're getting into higher salinity. And you can see that the pink line is last year's data. The pink line is 2024. So there's a lot of variability in what anchovy are doing in terms of their relative abundance, and also kind of what salinities they're found in, but they tend to be much more abundant in salinities of 20 to 30, right, in saltier water. So if we break this out and we look at, okay, how does that abundance change as a function of like critical dry, below normal, above normal years? You can see here that they don't really respond necessarily to outflow in those ways, which kind of makes sense. They're a marine species, but you do see that in wet conditions, they tend to be much more abundant in lower salinities, right? So that's when potentially the San Pablo Bay is much more fresh, and so they'll be there as a result of that. Interesting last year. I don't know whether last year was a above normal or a below normal year. I think it was just, it's something that I think people are having a difficult time characterizing. And so that's why we called it an abnormally normal year. But in any case, that's what it looked like. So this is now I'm going to kind of get into the sonar part of the story, right? So we have these echo sounders that we have mounted on a pole that we deploy beneath our boat, and we go out in the nighttime, go out in the daytime. Most of our daytime stuff was associated with the Bay study to try to validate, maybe compare what was actually caught in the nets relative to what we're seeing on the echograms. How are we doing on time? Close? Okay, 5 minutes. Okay, so this is what that looks like. And so, unsurprisingly, we're seeing a lot more anchovies in the echo sounder data than we were seeing in the net samples. And so, here's May and June. We were only able to get the echo sounders to work for May and June. So that's what that looks like. And so in the light green bars are the Bay study trawl data. And there's no error bars with the Bay study trawl because you catch what you catch right in that moment. But with the echo sounder data, we're actually binning it all the way through the water column. So you can kind of get an estimate of abundance over that period of time, over a 12-minute trawl period. One of the things that we did to try to look at what was going on behind that wide variation that we're seeing with the echograms was to simulate an oblique trawl like what we were seeing with the Bay study, right? So this is actual data following one of those 12-minute trawls, and it's integrated over 2 meters. And so what we did is we just looked at the abundance of anchovy that were observed on the echogram along this diagonal line in 2-meter depth bins going all the way up to try to see how well that matched with the Bay study catch data. And you can see here that it didn't really match very well at all. The dark blue bars are essentially what we observed, and then these greenish ones were the simulated trawls, and then here's the net catch data. So I think what's interesting about this, obviously this is just 1 year of data, but it's indicating that there's really a lot of variation in anchovy distribution that's not being captured by these net samples, perhaps. And so we need to kind of explore this a little bit further. Just to give you an idea of what our night sonar transects look like, rather than being tied to the Bay study boat, we were able to just run these really long transects from bottom salinities near 20 to bottom salinities near 5. So in May, this is kind of what that looked like. The size of the circles represent the relative number of anchovies that were observed on the sonar. You can see that predominantly they were out in San Pablo Bay. It's important to look at the scale bar here. So, these are, we're up in like 700 to 874. What's the measure here? I think it's I think it's catch per 1,000 meters or something like that. I'm not sure exactly yeah. So anyway, the scale bar is much smaller here, but we were detecting anchovies in Carquinez Strait in June. So as that salt wedge moved further landward, the fish did as well, but in much smaller abundances. So then this whole depth question, right? So at nighttime, are they potentially more exposed? Are longfin smelt more exposed to anchovies in the water column? You can see here that anchovies tend to distribute much higher in the water column at nighttime. You can see that a little bit in June. We just saw very few fish in June. And I'll wrap it up. This is what an echogram looks like. And this is the same period of time. This is over a 5-minute interval though. So you can see these box plots kind of represent how anchovies distribute relative to depth. And you can see that they tend to be higher in the water column. Again, in June, much lower numbers, but the same kind of pattern held up. And then in the daytime, the same thing, right? These are all the Bay study stations that we went to. This is station number 345, which is down here in deeper water. And you can see that these fish were still higher in the water column when in the daytime we were observing much higher numbers of longfin smelt deeper in the water. So in summary, longfin smelt are more abundant in the lower water column where there potentially may be fewer predators. And the sinking behavior may be taking advantage of gravitational circulation to stay in the low salinity zone and not be in San Pablo Bay where all those predators are. That's a model that we're going to be working up with WIM going forward to try to look at clearance rates and see potentially what kind of impact the relative abundance of anchovies might be having on longfin smelt. We saw that the copepods are more abundant when X2 is low or under high outflow conditions. And that the anchovies were much more abundant in higher salinities. But in wet years, there may be a potential more overlap with what we're seeing with longfin smelt. All right, so yeah, with that, anchovies are way more abundant in May and higher in the water column. And for our next steps, what we're going to be doing is looking at March, April as well. We're going to be running these gridded transects, and then trying to capture what's happening with anchovies over the bathymetry profiles. The longfin smelt trawl will be an oblique midwater trawl rather than a surface trawl, followed with a deep trawl. And I think that's pretty much it. We're going to try to capture more salinity measures over the transect period. So with that, I'd like to thank everybody that helped out, state water contractors for funding, Department of Water Resources, permitting agencies, so on and so forth, and a large number of people that helped in the field and the lab. Thank you.
All right, thank you. I don't think we have time for questions, but feel free to find him at the break. So next up, we have Cristina GB Law from Southern Illinois University.
Hey guys, thank you for joining my talk today. I'm Cristina from Southern Illinois University, and the research that I will be talking about was in collaboration with UC Davis from the Dr. Sadros group, as well as being funded by the Metropolitan Water District of Southern California. And so previously, last year, we conducted a study and published the findings on baseline contamination concentrations found in the Sacramento Deep Water Ship Channel. And through that study, we saw that there was consistent contamination across 6 different sites that were sampled longitudinally. The highest concentrations were found in the suspended solids, mostly associated with polycyclic aromatic hydrocarbons, or PAHs. When we were in the middle of doing the sampling, we often saw large cargo ships passing through the sites as we were going through. And we visibly saw how much more murky the water would become after the ship passage. And so this got us to sort of think about how this ship-induced turbation essentially would impact different species in the water column and how they would be exposed to contamination from it. And so the study that I'm going to be talking about today is showing how turbidity or total suspended solids were impacted by the ship traffic, as well as if ship traffic posed any additional risk on contaminant uptake or exposure to aquatic organisms. And so in June of 2023, we went back to the channel. We've sampled channel markers 77 up towards that West Sacramento port. And at that channel marker, we sampled when 3 ships passed through before and after. We collected turbidity and total suspended solid measurements, and we also collected suspended solids, zooplankton, and sediments to extract their matrices in lab to go and identify 66 different hydrophobic organic contaminants that were inside of the matrix tissues. What we kind of saw during our study was that I have a small simulation over here to kind of show that ship movement would keep suspended solids elevated in the water column. And so obviously, this is not to scale, but we would get a ship that would pass through, which would suspend some of the bottom sediments into the water column. And as time would pass by, all of the larger particles would start to go and settle back down into the bed sediments. But before the smaller particles had the chance or opportunity to go and settle back down, another ship would pass through. And so this would continue to keep the suspended solids elevated in their water column. And for Delta Smelt, elevated suspended solids aren't always seen as a bad thing. There's been studies showing that there was an increase in feeding success and survivability based off of using suspended solids as a cover. But when you look at it in terms of contamination, this poses a risk because of the elevated concentrations. So first, I'm going to show some of our data from the turbidity and total suspended solids. On the left-hand side, we have our turbidity measurements split between the before and afters for our box plots, and the turbidity is in NTU. In both, or in all three of the cases, the median was significantly higher after the ship would pass through, and the distribution shape was significantly different. For the total suspended solids on the right-hand side, with the bars being before and after, and the y-axis being total suspended solids, it increased after each of the ship passages, but significantly after ship 2. And these differences could be differences in the sampling time. It could be differences in the boat length or height or loading conditions. But overall, from all of these, it showed that there was an increase in turbidity and total suspended solids after ship passage. With this increase, again, with the terms of contamination, right over here on the left side, I have the contaminant concentrations. The two columns are split between the before and afters. The x-axis is showing the differences in the matrices, and the y-axis is the concentrations. In all of the polyaromatic hydrocarbons and organochlorine pesticides that we sampled, there was significantly higher concentrations in the suspended solids than in any of the other matrices sampled. For the sediments and the bioaccessible sediment concentration predictions that we got into the lab, these are not able to be seen on the scale for the organochlorine pesticides just because of the sheer magnitude higher of the concentrations in suspended solids. And also for those differences in pyrethroids, they're found in the sediments and the bioaccessible sediments, but not in the water column, possibly due to the seasonal inputs of pyrethroids being in the springtime rather than the summer. But all of this is basically showcasing the elevated concentrations in the water column, either within the zooplankton tissues or the suspended solids. There was no differences in the concentrations before or after the boats would pass through, which showcases that there is consistent contamination being exposed to those pelagic organisms. On those pictures that I have on the right side, it's showing that before and after from the boat would pass through. And again, you can visibly see the murkiness of the water and the elevated contaminant concentrations. And when they are exposed to these contaminant concentrations, there's a higher likelihood of the uptake of the contamination into the organisms, whether from ingestion, inhalation, desorption and reabsorption into the body of the fish through their gills from inhalation. And so all of this elevates that exposure, which could lead to adverse effects. Within a study from Hammock in 2015, approximately 60% of the Delta smelt in the deepwater ship channel were caught with lesions. And this was associated more so with contaminant concentrations rather than any other types of exposures. And then there was another study that was conducted in 2002, which showcased that there was a threshold for increased injury, including lesions, reduced fecundity, inhibited growth, which that threshold for the total pHs was 1,000 micrograms per kilogram dry weight. And just to put it into perspective, all of the suspended solid concentrations for the total pHs that we found were from 4,000 to 9,000. So that is a 9-fold increase in that magnitude from that threshold. And so again, showcasing that concern for the uptake of contaminated solids in the deep water ship channel. I would like to go and take this time to again thank our collaborators from the Dr. Sadro's lab at UC Davis, as well as Dr. Acuna from the Metropolitan Water District of Southern California. Thank you.
Thank you. I think we have time for one quick question while we switch the slides over, if anyone has one.
Hi, I don't know if I just missed this or not, but when you were talking about the fish or the ships passing, there was one ship that would pass and then suspend the stuff and then another ship. How often were the ships passing? Like what was the time between the ships?
Yeah, so for the time between the ship passages, we had our study be throughout a 7-day period, and we calculated that the average time during that period was 2.25 days between passage. When we compared this to data from marine traffic as well as from the USGS data points for the closest near that channel marker, it also roughly averaged around 2.65 days.
All right, thanks again. Next up, we have Lex Thomas from the USGS.
Can I bend it? I don't know, I'm afraid to touch it. Okay. Hello, my name is Lex Thomas. I work for the United States Geological Survey, specifically the hydrodynamics team. We're located in the West Sacramento project office, and today I'll be sharing with you our long-term continuous flow monitoring in the Sacramento-San Joaquin Delta. Our flow network has been gathering continuous flow monitoring data since the 80s, beginning as a small research group looking to develop measurement techniques for flow in an estuarine environment, and building towards an established project office. Today, our work has defined processes and methods used nationally, even globally, and has established the robust monitoring network that you see today. So we have 36 permanent surface water sites in the North, Central, and South Delta. These measure water level, velocity, and discharge, to name a few parameters. And this is being done with acoustic sensing technology, as you can see in the second photo from the top. This was first used by our office in the 80s. It was researched and developed over the decades and now applied to all of our gauge stations in the Delta. This is how we measure discharge in a complex tidal environment. And we also have water quality at all of these stations. This is done in collaboration with the Department of Water Resources, as well as our group and other USGS groups. This surface water and water quality data comes in every 15 minutes, so at a pretty high resolution. And today, to best illustrate how our data can be applied, I'm going to tell you about a case study. And to do that, I'm going to take you back in time to the 1990s and early 2000s to Frank's Tract. Frank's Tract is a flooded island. It's been flooded for the last 80 years. It's a large, predominantly shallow body of water that connects the central delta to the southern delta. It is located just north of our state and federal export facilities. And the hydrodynamics of this area are very interesting, as well as it's in proximity to key infrastructure. But despite the interest in Frank's Tract, at the time, there wasn't that much large-scale data here. But thankfully, the hydrodynamics group had a way to measure for this data in a tidal environment. So how did our network look at the time, and how is it applied to Frank's Tract? At the time, we had 13 stations dispersed throughout the Delta. They were using experimental science to collect high-resolution surface water data. This is that acoustic sensing technology I mentioned earlier, and it was looking really promising to apply to more stations throughout the Delta. And although we didn't have any permanent stations around Frank's Tract at the time, we were participating in collaborative science in the area. This figure is from one of those studies in the 2000s. The rounded dots show temporary stations for both surface water and water quality. And these temporary stations were not only useful to that study, but they were really useful to looking into the hydrodynamics of this area. And they were really highlighting the way this experimental science was working at the time. It was really highlighting a big gap in data that existed around this area. So resource managers and our group were in agreement that these data gaps needed to be filled. So in 2006, the Bureau of Reclamation funded the Central Delta expansion of our stations, and we had 4 stations permanently installed around Frank's Tract. So how did these permanent stations benefit Frank's Tract? They led to high-resolution data collected from the monitoring network, provided a detailed look at the tidal cycles, tidal fluxes, and unique events happening in this area. This is a great improvement upon having no data or point data that was available in the area. And ultimately, it led to a better understanding of the hydrodynamics in Frank's Tract, namely salinity intrusion, which is a huge problem for this area. Having 15-minute data also led to more informed real-time decisions. And the data was used to calibrate models regarding Frank's Tract. This is really important for resource managers, especially in regards to the salinity intrusion for potential drought scenarios. And I keep mentioning salinity intrusion The data that was collected at these stations can help us visualize that salinity intrusion. So on the left, we have visually interpolated data from our water quality stations throughout the Delta. And on the right, we have a drought model that shows a drought scenario in the Delta that was calibrated with our long-term data. Both the real-time data and this drought model show salinity intrusion into Frank's Tract. So in those top images, the magenta and red being higher concentrations of salinity show salinity moving in from the bay and then going into Frank's Tract in this salinity intrusion. And the drought model, you can see in the second image that, of course, salinity gets worse as the drought worsens and it gets a higher concentration in Frank's Tract. And then at the bottom, when fresh water does finally enter the system, we get a first flush event. That salinity clears out through most of the Delta. However, it remains in Frank's Tract, and there's this delayed withdrawal of salinity from that area. This high residence time of salinity can have negative effects on the ability to export fresh water from the Delta. This salinity is really what underpins that availability of fresh water. So it's really important to understand the salinity intrusion. And this exact scenario in drought became relevant in the 2010s and 2020s when there was drought in California. Having real-time data and these models were able to inform management action to prevent the salinity intrusion into Frank's Tract. So this picture is of the salinity barrier that was put in place by the Department of Water Resources. And what's cool about having a long-term monitoring network is when actions like this are taken in the Delta, we then have stations that can monitor the impacts of these stations, of these actions in real time, as well as look back on them retrospectively. So this is just one case study located in Frank's Tract, and the data and applications that were made with this were all possible because of 4 stations in the Delta. Now, the same data possibilities and benefits that exist for Frank's Tract exist for all of our stations today in the Delta. So again, we have 36 gauge stations throughout the Delta that measure for surface water parameters and water quality every 15 minutes. And so a wide application of our data does exist today. From the top of the images to the bottom, we have restoration happening in Lookout Slough that we are monitoring, as well as salmon out-migration survival studies, how the hydrodynamics plays in potential harmful algal blooms. And lastly, but most importantly, a lot of our stations are indicators and show the current conditions moving through the Delta at the time. This is relevant at our Freeport station, for instance, for when the Sacramento Sewer District can release reclaimed water out into the Delta, depending on the tides. And as mentioned in our plenary, the ODM stations, and OBI and MDM and our USGS gauge stations, those help indicate flow going into the Old and Middle River. And lastly, long-term monitoring data serve as a frame of reference while assessing the impacts of proposed actions here in the Delta. This is more important now than ever, given that we live in a world with a changing climate and that more people rely on the Delta now for their resources. So I would invite you all to look at our data if you found this interesting. The National Water Dashboard is where USGS leaves its QA/QC data and approved data. Bay Delta Live is a multi-agency collaboration that provides our data in a provisional manner. And this California Data Exchange Center, run by the Department of Water Resources, has our provisional data as well. Big thank you and acknowledgement to our stakeholders and collaborators, because without their contributions, we would have no flow network today. And also a big thank you to our hydrologic technicians, because without them, we would not have these stations reporting data day in and day out. Thank you.
Thank you. I think we have time for one question. Yeah. Last call. Nope,
Nope.
there's one.
I believe the first site was put in at Freeport. That was a Freeport gauge station. And I'm looking at my colleagues who've been here a lot longer. I believe that was put in the 70. I'm not gonna get into years, but that station has been reporting gauge data since the 70s. 57, I'm being told. Not acoustic, but data since 57.
All right, thank you again. Next up, we have Tara Morgan King, also from the USGS. Yeah.
Okay. And then, all right. Thank you. I'm Tara Morgan King. I'll be talking today specifically about the exciting wet year of 2023 with relation to our Delta Suspended Sediment Network. I am from the USGS California Water Science Center. And I want to highlight the importance of atmospheric rivers. Let's see. Sorry, I'm getting my orientation here. Atmospheric rivers are important to California. The role of atmospheric rivers during 2023 was really significant in terms of flow and sediment into the Delta. And there was really significance in terms of flow and sediment coming from the McCallamy River watershed, which was specifically interesting because the McCallamy River watershed is normally, in terms of sediment contribution to the Delta, roughly about like 3% of the total sediment load coming into the Delta during kind of like normal conditions would come from the McCallamy River watershed. And the importance of, I'm going to also describe the importance of the sediment in terms of managing our 12 FMU turbidity limit in the Delta at various stations. California has the largest precipitation variability in the United States. You can see the map on the left shows California, the variability there, highlighting the colors there, lots of variability compared to the pink dots, which is very low variability. And so that's attributed to the landfalling atmospheric rivers that we see, these narrow bands of concentrated moisture, and they specifically contribute to the annual precipitation totals. The map on the right shows that fraction of AR contribution to the total precipitation that we get in California. So specifically the trajectory of atmospheric rivers coming to the West Coast, they're more predominant in the north, Washington, Oregon, and will provide more moisture to Northern California than Southern California. And this is an example of that, 43 degrees latitude compared to 38 degrees latitude. The bar graph shows a total number of atmospheric rivers counted from 1960 to 2024. And the bars on the right, the red bars show this difference in 43 degrees versus 38 degrees in category 5 atmospheric rivers. So atmospheric rivers, this terminology, Pineapple Express, they have, you know, they're not a new, they're not new to our science, but specifically categorizing them is a newer, it's been established within like the last 5 years out of the Center for Western Weather and Water Extremes in San Diego, at Scripps Institute in San Diego. So there's a categorization from not an atmospheric river, it's just a storm that doesn't qualify in terms of the integrated water vapor transport, up to 5. So there's a categorization, category system that was developed similar to like a nor'easter and a hurricane. So specifically, these atmospheric rivers account for 85% of the multi-year precipitation variability in California. So it's either a drought in California or it can be a matter of extremes like we saw in 2023. So almost half of the drought busting storms are from landfalling atmospheric rivers. So this is showing a comparison. The map on the left is showing like eight landfalling atmospheric rivers categorized as strong. Only 8 for a combination of 3 years. So our drought years, 2020, 21, 22, compared to 2023, where we had 7 instances of strong or greater magnitude. They can be extreme or extraordinary. So that's what we saw in 2023. And you can see, I want to point out the strong landfalling atmospheric rivers spanned California. So we also saw that the landfalling atmospheric rivers spanned our range of our watersheds going into the Delta. So the Sacramento River, the McCollum, and the San Joaquin. So suspended sediment monitoring and sediment into the Delta is specifically important. It creates and sustains the Delta landscape. It's important for habitat suitability. It provides a source of nutrients and of course contaminants. We use it to assess ecosystem function, as you've seen in multiple other talks. It's necessary for tidal wetland restoration planning, ship navigation, sea level rise. And the primary source typically, as I've explained, with the landfalling atmospheric rivers and the trajectory to California, that they typically, it's more common for them to hit Northern California. This is a graph of our Freeport station, which is our northern boundary of flow and sediment coming into the Delta. Sacramento River and the Sacramento River watershed provides roughly during normal conditions, 80% of the flow and 80% of the sediment into the Delta. So that's what we typically manage for. And in terms of sediment, there has been a decline over decades. And so currently where we are with total sediment coming into the Delta on an annual basis is 1,000 metric tons per year. So I want you to remember that. And we can look at the future of sediment transport. This is modeled. This is just an example of our colleagues in the USGS California Water Science Center talking about climate change and implications for suspended sediment and flow coming into the Delta. They described conditions from 2010 to 2019 applied to global climate models. And their results found that there was actually an increase in the trend over time through 2099 of suspended sediment. And this is attributed to more high flow events than historical that can be also attributed to atmospheric rivers. So this is the reason why they have found an increase in sediment loads. In 2023, we saw multiple landfalling atmospheric rivers at the very end of December 2022 into the very beginning, January 1st, January 2nd of 2023. This is a visualization of turbidity, and this is January 2nd, 2023, where the turbidity just skyrocketed all over the Delta. This is related, obviously, to maintaining our turbidity stations that are monitored to avoid the turbidity bridge at OBI, looking at the integrated early pulse protection at Freeport, and hoping to avoid any OMR actions. And so the questions for 2023 were what effects were related to the exceedance of, or what effects are related to exceedance above 12 FNU. A role, if any, of clear water from reservoirs, larger sediment loads from the McCollum during wet years, larger sediment loads from the San Joaquin in wet years, and what role of sediment transport into Frank's Tract deposition, subsequent wind wave resuspension. And so this is a result of what we did observe at those three turbidity trigger stations that are in the biological opinion. 12 FNU was exceeded at PRI, OBI, VCU during January of 2023, extended for multiple days, over a month. And so we saw that this was attributed to the atmospheric rivers and rare peak flows, which I'll show you in a couple slides. After this, the influence of the McCollum watershed, and we saw higher than average sediment concentrations. And so I want to remind everybody that turbidity is just a measure of water clarity. It's used as a metric. It's non-conservative and cannot be modeled. The suspended sediment monitoring network is important because it is conservative. It can be modeled. We use it to compute the direction and magnitude of transport. And turbidity is the result of the sediment concentrations, not the reverse. So we need to monitor the suspended sediment concentrations to be able to describe these turbidity conditions. So I told you to remember that 1,000 metric tons, that is our average annual sediment load coming into the Delta from the Sacramento River watershed towards the end of our declining trend. In January of 2023, we saw almost that in the matter of a month. It was even specifically, it happened in the matter of days at the very beginning of January. And so here we're looking at the peak flows from 1997 to 2023 at our Freeport station, which is the northern boundary of flow coming into the Delta. It was elevated above average, looking at a median flow of roughly 73,000 CFS. We saw a little bit above average in 2023. This is just showing the peak annual. Specifically, the McCollum River watershed was almost equivalent to what was coming in from the Sacramento River. So there was really rare peak flows. It was the third highest peak flow on record since 1907, since the USGS has been managing that gauge since 1907, and it was ranked as the second highest since 1967. The San Joaquin River was also above average. It was nearly 4 times the annual peak in January. So high flows were coming in from every tributary into the Delta. So summarizing that, typically the Sacramento River is always the largest, roughly 80% of the total coming into the Delta, and we can manage OBI based on what is typically coming in from the Sacramento River. But in 2023, if loads are high, like we saw in 2023, San Joaquin River can be important to VCU. And then the McCollum River adds to the loads already coming in from the Sacramento River and is extremely flashy. When flow comes in from the north, it typically goes down Georgiana Slough to, we observe that sediment and turbidity at the McCollum, our MOK site. This is an example of kind of a normal condition. This is in 2022, a first flush that we saw. And kind of normal conditions, the top graph is showing discharge. So our Georgiana station and our MOK station are similar. And then the SSC, the sediment concentration in the middle graph is showing that it's much higher at Georgiana Slough because the sediment is coming from the Sacramento River. And then the sediment flux in the bottom graph is showing much larger sediment load at the Georgiana Slough station than the McCollum. And so that's typical. As you reach the McCollum station, it's much more tidal. There's slack tides and sediment deposits along the way. So the map on the left is showing 10,000 metric tons measured during the first flush, as opposed to 4,000 metric tons at McCollum. So there's deposition along the way. And so with only 4,000 metric tons can be more easily managed in terms of what might get transported into Old and Middle rivers. As opposed to January 2023, we saw 140. So the last graph showed you 10,000 metric tons. Now we're showing 140 just in January. And then that was almost doubled by the influence of the McCollum River watershed. We measured it at MOK, it's 210 compared to what we saw at Georgiana. And the flow coming in from the McCollum, this graph, the top graph shows discharge at the Moke station versus GSS. You can see the flow at Moke in red is nearly 4 times that coming in from Georgiana Slough. So Georgiana Slough might see around 10,000 CFS coming in from the Sacramento River. Higher flows might be upwards of 15 or 17, but during 2023, with that influence of 65,000 CFS coming from the McCollum River watershed, it was really dramatic in terms of the advection that came down to the MOK station. And then the sediment concentrations were equivalent, where normally we see smaller sediment concentrations at MOK, and then the sediment flux was larger at that first part of January at the McCollum. And with those larger flows coming in from the McCollum River watershed, this MOK station, this graph in the right is showing the velocity. There was peak velocity. The velocity exceeded any instantaneous velocity since our MOK station was established in 2008. And flows became unidirectional. So that means that the flow was directed towards the south constantly over multiple days, and the sediment concentrations were pushed directly towards the west and to the south. So they went into the San Joaquin River and directly into Old River because of that unidirectional flow with really high velocities. And this is showing our OSJ station. That's a newer established station. So 28,000 metric tons of sediment estimated coming in during this time is really large compared to what we normally see. And then, so the flow coming at the McCollum directed down to our OSJ station affected the net flow and the tidal excursions, the velocities. And this middle graph is showing how I computed the tidal excursion based on our velocity data. And during the flood tide, the tidal excursion can be described as the distance that that parcel of water travels during a tide. So this line in the map on the left shows that that tidal excursion from that higher velocity went all the way down on a single tide past that OBI station. And really immediately the SSE increased in Old River. The bottom graph on the right is showing the sediment concentration at that OSJ station. So as I've said previously, that typically 80% of the flow and sediment coming into the Delta comes from the Sacramento River watershed. This pie graph shows the conditions during 2023 where it was coming in from every watershed. It was kind of equal. Of course, Sacramento River was still the largest contributor. But with that, in 2023, these bar graphs show the Sacramento River, the MOK station, and the San Joaquin River January average suspended sediment concentrations. So 2023 was higher than any of the other years, including 2017, with 2017 having higher flows and a wetter year. There was more reservoir releases associated with that. So that was part of it. But these graphs go back to 2013, so 10 years. Highlighting 2023 as larger. And then with that, we also saw record salmon return to the McCallamy River during 2023. It was the most successful in more than 80 years. This is a news clip from East Bay Mud that they documented in November of 2023, Fall Run Chinook. And previous to that, the record was 2017, but 2023 beat the record. So with a warming climate, extreme atmospheric rivers they're predicted by multiple atmospheric scientists to become more extreme. There's going to be a greater availability of water vapor in the atmosphere because of warming temperatures. So ARs will become more frequent, have a higher intensity, and heavier precipitation associated with these more intense atmospheric rivers. So thank you, and thank you to our amazing team. And then we have posters upstairs. So I invite you.
Great, thank you so much. Now for the final speaker of this session, we have Bryan Gopez from the USGS as well.
Hi, I'm Bryan Gopez. I work with the USGS Hydrodynamics Group, as you heard, in the West Sacramento Projects Office. And today I'd just like to share with you our long-term suspended sediment monitoring network, specifically in the Sacramento-San Joaquin Delta, and I'll go over a little example of how we use it in Frank's Tract. So this is just the outline of my talk. I'll show a brief overview of the whole SF Bay Delta monitoring network. I'll go briefly into the techniques and methods we use to collect and process our samples. And then I'll talk again about Frank's Tract. So here is a big blown up map of our whole SF Bay and Delta monitoring network. Just as a note, that line is not any specific line, it's just a general indicator of what we consider Bay and what is Delta. As another note, this talk will only go into the Delta. So if you are interested in anything about San Francisco Bay and our modeling, sampling efforts there, please come talk to us at our poster. So to start off, I'd like to give a little history of our suspended sediment network. And it started in the 1990s, mostly to measure sediment transport rate in regards to ecosystem restoration. These stations describe sediment inflow to the bay, as you heard from the Freeport station near the north, at Sacramento River, and our Stockton station in the east. These also describe transport, deposition, and erosion of sediment within the Delta. Then in the 2000s, it was expanded with the Upper Cache Sluice stations. These were put in to study the hydrodynamic effects of the Liberty Island for Delta Smelt habitat restoration. And this is a more modern picture of our station. Of course, not as comprehensive as the first big map, but as you can see, it was greatly expanded in the 2010s to support numerical modeling efforts. And as a note, these stations are often co-located with our flow stations, which I'll explain why in my next slide about techniques and methods. So some of our products when measuring discharge and sediment are, of course, our discharge time series and the suspended sediment concentration time series. We need both to be able to compute the suspended sediment flux and loads within a specific region with a specific time. And when we take our discrete samples, we use our discharge data from our onboard AZPs. We take a transect of the channel, and we then compute the channel and split it up into five cells where there is an equal amount of discharge coming in through each cell. This is to set up our equal discharge increment, where we then go to the centroid of each cell, sample the water column all the way down and back up with our US D96 depth integrated bag sampler. And the average of that gives us a depth integrated cross-sectional average suspended sediment concentration, which we then use with our discharge to compute flux. So going back to Frank's Tract, as you heard, is a flooded island, and it also acts as a sediment basin, which means that whenever sediment gets in there, it kind of stays in, settles down, and only gets resuspended and exported with with a wind event. That is demonstrated with these graphs. We have our input stations in black and red, Falls River and OSJ. Those are the inputs from the San Joaquin and the north into Frank's Tract, and the output stations in green and blue are all of the sediment being exported out of Frank's Tract into the South Delta. The three graphs on the right, starting from the top, that is our turbidity data taken from the Freeport station in the north. The middle is, of course, the four Frank's Tract stations the turbidity data taken from the four Frank's Tract stations. And the bottom graph is wind speed data taken from the Travis Air Force Base. As we can see with the top graph of Freeport, we see with any fluvial event, there is a spike in turbidity, and after a time lag, we see the turbidity at our input stations also spike up. And then again, after the fluvial event, after it all settles, it goes down naturally, but with a wind event and another resuspension and export of the sediment, we see in our output stations, they get a gradual increase to above the 12 FNU trigger threshold as mentioned earlier. And again, what's important is that this is a gradual increase, meaning that it will stay above or often stay above the 12 FNU trigger threshold over an average of about 14 days. So this graph is this is not a graph. This image is taken from Bay Delta Live just as an example of how we see sediment settling in Frank's Tract, because Frank's Tract itself is higher in turbidity units than the surrounding, and how the outputs then start to come up along with it. Of course, this is just one way that our data could be used. And if any of this seems interesting to you, or if anything in the Bay is of interest to you, I urge you to take a look at our data online, or come talk to us in person or at the postering session. And again, once again, we'd like to acknowledge all of our collaborators, because without them, we wouldn't have a sediment network. And thank you for listening.
I think we have time for a question or two.
Yeah, thank you for your talk. So when the settled sediment gets suspended again from the Frank Tract, you said they leave like to the southern stations, like toward the South Delta. Do you know how far into South Delta the suspended sediment travels to?
how far it traveled into the South Delta, right? Well, we see effects of it all the way from Frank's Tract into the OMR stations, the OBI and Middle River. And the South Delta stations like VCU, like that whole Tracy area. Just, yeah, it mostly depends on the flow and how far it goes, but we can see it like down there, right? In more like the Tracy area. H4, Old River Highway 4 and like that area.
Thank you.
Thank you.
All right, well, thank you so much. Thank you to all the speakers for the session.
All right, we'll go ahead and have our next break. Please be back at 11:10 for session 8.
Hi there, welcome to session 8, fish modeling. My name is Matthew Murphy and I will be moderating this session. First up, we have Chase Ehlo with Central Valley Steelhead.
Cool, okay. Thank you, Matt. I'm Chase Ehlo. I'm with the U.S. Bureau of Reclamation in the BDO office right here in downtown Sacramento, and I'm going to talk about a framework for a juvenile production estimate for Central Valley Steelhead. Just want to thank my co-authors here. They've all contributed in some way, whether that be writing or data or helping with analysis. So let's get started. So I'm not going to name every individual here, but these JP efforts really started back in around 2021 with the workshop and then a science plan that got finalized in 2024. A lot of people, agencies, organizations involved in this, and this list is probably not inclusive of everybody that's been involved. Oh, okay, so to talk about steelhead, you really have to talk about rainbow trout in general, or omicus as we like to call them here. They're very widespread and abundant in California, as most of you know, and they're also widespread in the Central Valley. This is a species that has a lot of different life history expressions, something over 32 now. I think they keep adding them every day, I feel like, but the anadromous life stage is the one we're going to be talking about here, the steelhead, and that is federally listed as threatened. So there is a suite of monitoring programs, again, as most of you know, in the Central Valley. These are generally designed for Chinook. They sometimes catch Omicus, but they're not really keyed towards Omicus. So things like rotary screw traps, trawls in the Delta and whatnot are pretty inefficient at capturing the species. So because of this, let me see, got to find my spot here. So because of this, we don't really have a good monitoring program for Omicus, and then take limits such as loss in the export facilities in the Delta aren't really tied to a population metric. So this really highlights that need for a valley-wide life cycle monitoring program. So this spawned that workshop that I mentioned earlier, which resulted in this stillhead science plan. You can find it in the QR code here. I've got this at the end of the talk if you don't have time to scan it here. So this was designed to better inform those management decisions of the CVP and SWP water programs. So they wanted to do this by accelerating that research and monitoring of the Central Valley stillhead and to ultimately inform that stillhead juvenile production estimate. So the purpose of this talk is twofold. I want to discuss that JPE framework and demonstrate it in the Stanislaus River. And then I want to discuss some of the shortcomings with the framework and some of the next steps. So you may think we have a JPE for winter run Chinook. How hard can it be to develop a JPE? Well, it's pretty hard. But a Chinook life cycle is very circular. I'm sure everybody knows this. They spawn, they outmigrate, they come back, they spawn again, they die. And it's just a circle of life there. With the Central Valley stillhead or Omicus, you start introducing all those life history variations and it becomes a lot more complicated. So you have a cycle that looks more like this, where they hatch and they grow. They can reach age 1, decide to outmigrate. They can stay residents till age 2 and then decide to outmigrate. Sometimes they spawn and then outmigrate and come back and spawn. So there's a lot going on here. So calculating a true JPE for stillhead can be very complicated and resource heavy. So the science plan outlines a couple of different methods for simplifying this approach. You can read about that there. I'm not going to go into too much detail about that, but I'm going to focus on one today. So this approach focuses on estimating the number of age 2 smolts outmigrating through the Delta. That's believed to be the predominant age class that's outmigrating. So what we're trying to do here is cut out all these complex life stage transitions and solely focus on how many fish, regardless of age, regardless of life history status or variation, are spawning, how many eggs they produce, and how many of those fish outmigrate as age 2 smolts. We call this the adult approach, and this is a 2-year forecast. So if you have a spawner abundance for 2023, that would be a forecast for JPE for 2025. So this is the actual equation that's in the stillhead science plan. Essentially, it's a giant conditional probability statement, but you start out with a measure of spawner abundance and egg production, then calculate these life stage transitional probabilities that I have listed here. I'm not going to go into too much detail on that. But I wanted to try to apply this equation to some real work data on the Stanislaus River. So in green on this table are numbers that we're actually getting from projects we fund in the Stanislaus. This is mainly from Kramer Fish Sciences work. So that's in this report. So we get all 2024. And in the red or orange, depending on what you're seeing on the screen behind me, that's data that I just kind of found in literature. So we're just calling these surrogate numbers. So this is this is some Pacific Northwest stillhead stuff. This is Klamath River stillhead stuff. And then there's some Atlantic salmon stuff that exhibits some similar life history variation. Something else I want to note here is the female spawner abundance that I have up at the top. That's a 2022 estimate because I am using a 2024 outmigration rate from telemetry work in Stanislaus. So this JP I'm about to show will be a 2024 JP. So for the calculation itself, we opted for a Monte Carlo simulation with 500 iterations. So for each iteration, we sample female abundance numbers from the Stanislaus River. We assigned each of those female spawners a fork length based on some age and growth distributions in the Kramer findings. Then we calculated the total egg production for each iteration. So we end up with 500, a data frame of 500 egg productions. And then we worked through these probabilistic parameters, and we did this by sampling from these log normal distributions. I constructed these from the estimates that you saw in the table on the previous slide, and we end up with a data frame of 500 JP estimates, which looks something like this once we run the calculations. So these estimates range anywhere from 0 to 350,000. That doesn't tell us much, not very informative, so you can kind of summarize this into medians and first and third quartiles, and you get something around 25,000 fish for the Stanislaus River. So that's great. We have a JP there, but what does it mean and where do we go from here? Well, to make this meaningful, we have some current shortcomings of this for making this meaningful. So we have to scale this JP outside of the Stanislaus to other San Joaquin tribs and the Sacramento River. And as mentioned earlier, there are some data gaps. I was using some surrogate values for some of these parameters. So some of these data gaps are around early life history traits like fry to par survival or par to smolt survival. And there's also some questions about the feasibility of collecting some of this data year in and year out for the end of time. So I wanted to explore both of these shortcomings, show how I might address them in a really quick and crude way. So first we could talk about scaling. So this is assuming that you can only rely on Stanislaus data in the near future. I came up with a really basic scaling method that uses habitat area and a number of tribs in the Central Valley. So this is based on habitat area developed within the CVPIA program. So it includes estimates of spawning and rearing area at different stream charges. It also includes these estimates for fall run, spring run, and stillhead. But for the sake of simplicity, I'm just averaging the habitat area across different stream discharges and across every species. So I'm just disregarding the species because it was pretty kind of a shotgun approach on which species was modeled where. And once you do this, you get around 14 streams that range anywhere from 60,000 square meters in the American River all the way to 105 square meters of rearing habitat in the Bear River. And then I worked to relate the Stanislaus JP to the habitat area, to the habitat area in the Stanislaus. So I calculated fish per square meter for spawning habitat and rearing habitats. That would be spawning density and rearing density. Now I reverse calculated those for each trib based on their habitat area. And then I summed everything together to get a total JP, which looks something like this, almost looks identical to the other graph. The x-axis is in millions, but you can see that it's still a pretty decent level of uncertainty in the JP with this method, but you can summarize that again into that median, and it's about 1.2 million fish. If you're interested about what this means as an estimate in 2024, most people probably know that loss at the export facilities was exceedingly high in 2024. It was something around 5,300 fish. So this would be about 0.4% of the total loss at the export facilities. So if this JP was rooted in any kind of reality, then those Delta export operations likely had minimal impact on outmigrating steelhead. This came with many assumptions, as you probably know, because like I said, this is a very crude approach. This is assuming equal survival, equal outmigration rates, equal Delta routing probabilities. Also doesn't include every steelhead stream. But I really felt like this is maybe a good jumping off point for thinking about how to scale this up, and we could really dial this in with some more elicitation and thought. I also wanted to examine those JP parameters through the lens of a really basic plasticity analysis, so I can determine the level of influence each parameter would have on the JP. So to do this, I defined the range of each parameter, and then I would, for a single parameter, I would incrementally increase the values and keep the other values the same for the other 6 parameters, it looks like. Then I would iterate through all these parameters and summarize the data. Got to click. So that looks something like this. So if you look at the response of JPE to the parameters, you can see these slopes are steepest on H2 outmigration and spawner abundance. So that would suggest that those two have the biggest impact on JPE. This is a similar graph. This just shows the range of JPEs, min and max for each parameter when you do that. So ultimately, this is just a way to illustrate that these parameters may be more important as I framed it here. In this case, we may want to focus on H2 outmigration numbers and spawner abundance. Again, this, like everything in this talk, this overly generalizes everything in the JPE parameters, and it could really benefit from an expert elicitation, just getting some experts in the room to talk about what parameters are most important. You know, they may decide that H2 outmigration is the most important because that would have the most year-to-year variation. I don't know. But we'd also talked about maybe doing a more robust sensitivity analysis and looking at how these uncertainties in each parameter would affect the uncertainty of the JPE. So that's kind of a next step. So in conclusion, the JPE calculation itself is pretty straightforward. It's transparent. It incorporates those uncertainties and presents them. But really where we're running into roadblocks is informing these parameters because there's a lot of them and there's a lot of streams and scaling those up to a basin-wide result. Like I said, the scaling that I did was pretty messy. Came with a lot of assumptions, but it's a good jumping off point. And that simple plasticity analysis that provide us some insight on those parameters could be the next step in learning how to scale and deciding what life specific stages would be most important for monitoring or scaling this up. Just some next steps. We're going to continue integrating the Stanislaus data into the JPE framework. We've, per the new LTO and the ITP, there is a stipulation there to create a stillhead technical team within the adaptive management program, which we already started. And this is a interdisciplinary team, an interagency team that includes some federal agencies, state agencies, local agencies, and some NGOs. So I'm not going to go into too much detail on that, but we have some general short-term goals and long-term goals. Some short-term goals is we're going to have a Delta Science program peer review. I think that's going to happen sometime in late 2025. So they'll be able to go over some of our products and give us some feedback on that. We're also developing a hatchery surrogate program in lieu of a basin-wide JPE. This is a temporary fix to this problem. Some of the long-term goals will be addressing some of those data gaps and scaling strategies that I've talked about. And ultimately, this will all feed into a broad implementation strategy for the Central Valley and give us some kind of population metric for those take limits. So that's all I got. There's that QR code again if you want to access the Stillhead Science Plan, and I'll take any questions if I have time. A few minutes for questions.
Good talk, Chase. Thank you. I had a question, maybe I missed it. When you're coming up with the JPE for the Stanislaus River, did you factor in the probability of those basically undecided fish, for lack of a better term, to actually mature as an anadromous individual versus a resident individual?
I was just using, besides the surrogate numbers for the H2 outmigration, I was using the telemetry data. So that's the probability that those fish outmigrated or stayed, but it didn't include any kind of survival estimates on whether they stayed because they didn't quite get the data for that. So I don't know if that answers your question. Okay.
Got time for one more.
We have one from Laura. The winter and JPE is based on the JPI, which is a measurement at Red Bluff Diversion Dam. Are there no points in the system where you could use measured data instead of this estimation method, which has so many input parameters and uncertainties? Or are the monitoring data not good or specific enough for this to be done?
I think it's certainly possible at the Red Bluff Diversion Dam if you're interested in the JP of the Upper Sac. That would require us to figure out some capture probabilities for steelhead. I don't think they currently have those at the Red Bluff Diversion Dam, but in the San Joaquin, none of the rotary screw traps, or very few of them, get enough data to do that, it seems. The Caswell trap in the Stanislaus got, I think, did they get 0 or 2 fish last year? And then we just showed here that there was quite a few fish that outmigrated. So I just don't think most rotary screw traps are efficient enough for that, but I think it might be possible at the Red Bluff Diversion Dam. Thank you.
Thank you, Chase. Next, we have Gonzalo Castillo from the Fish and Wildlife.
Thank you. I would like to acknowledge my co-authors, Stephen Slater from California Department of Fish and Wildlife, and Bryan, U.S. Bureau of Reclamation. This is the third follow-up on study to trying to synthesize the information in the 20-millimeter surveys for early life stages of Longfin smelt and Delta smelt. So a lot of the methods are similar to the ones presented earlier in the science conference, so I will not go into much detail. There is. So the classical view is that these two species move downstream from their spawning areas. And also we know factors like water quality, temperature, turbidity can influence their occurrence. And on Longfin smelt also, we know that when we have wet years, spawning also can happen in the lower estuary. So that part of the life cycle is not considered in this presentation, since the 20 millimeters only surveys or monitors the early life stage up to the San Pablo Bay, or in this case is the North Bay in the map. Another clear things from past studies, feeding studies on these two species, there's a high variability, temporal spatial variability of prey. And the first analysis we conducted, the two first items that you see there, Pseudoadaptumus and Tortanus, appear relevant for Delta smelt, and the lower two, Limnoitona and Eurythemora, appear important for Longfin smelt. Keep in mind that a lot of these are introduced species, so that reiterates the tremendous change in the food web in the upper estuary. Not least is the climate change evidence through the severe droughts that we have had. As you can see in this, this give another support for try to analyze these periods in the entire period of the data set from 95 to 2017 into different periods to try to see if there's better associations within periods. So you see that both temperature and salinity show an increasing pattern across areas in the estuary and across years, increasing in more recent years. So the objectives were to identify potential biotics and abiotics drivers for the abundance of delta smelt and Longfin smelt in the upper estuary over these 3 periods. The baseline, abbreviated from now on as BL, from 1985 to 2001. The pelagic organism decline documented since the early 2000s, but extended up to 2012. I'm using a Temple et al. Reference for this periods that they did a review on Longfin smelt and delta smelt, so on the climate change intensification period, beginning in 2013 and ending in the period of analysis for this presentation, 2017. So for the analysis, we have 10 areas for the stations, and there are 51 stations assigned across the areas, and you can see there the X2 position, 45, 85, 75, 85, and so on. And the survey is conducted from a spring to early summer. Luckily, we have no missing years. Up to 9 surveys are conducted every other week. So this is the gear. It's a very rare opportunity to find a gear that includes both some of the explanatory variables. As you see in the top of the net, you have the mesozooplankton net that concurrently collects zooplankton, and the bottom larger, the larval juvenile fish net with a much larger mesh size. And each of the toes include oblique toes, so from near the bottom to the surface. And for the mesozooplankton net, one toe is analyzed per station, as opposed to three toes per station for fish. In terms of length composition, there's similar distribution for both species. The primary mode being around 12 to 21 millimeters. So another consideration is that for the analysis, we only include larval and juvenile stages, so individuals larger than 55 or 90 millimeters are excluded from the data set. So in terms of methods, non-parametric multiplicative regression was used. And so the response to environmental gradients and interactions among explanatory variables is the goal, to try to identify among thousands of models, potential models, the one that have the highest explanatory power. And being a free distribution-free method, one of the attractive things of this modeling is that the variables are multiplicatively added instead of additively added. So when one variable, one condition is extreme for survival and there's 0, then in the predictions that is considered 0 output rather than being a function of other variables that could show presence of or abundance in that area. Model selection, they were cross-validated by using the XR square, and the improvement criteria used for selecting was a difference of at least 5% from the previous model with less variables. Sample units included the area and all the surveys between March and July and year. So it's a combination of area, survey, and year. And again, the periods analyze baseline, pod, and climate change intensification. For response variables, the catch per unit effort, number of fish per 10,000 cubic meters, log transform, explanatory variables. We have water quality, 3 water quality samples from the survey. They also include turbidity, but consistency, use SECI since that was originally included. So plankton, we have 22 taxa, different life stages of copepods and general stages for cladocerans, arpacticoids, and rotifers. Special variables, area, latitude, longitude, and bottom depth. So we'll see the model outcomes for each of those periods for each species. So the table here and the graphs on the top of the table, I want to point out the model with the highest number of variables is the one that is used to show this surface response plots. And you see the variables that are included in the models, in this case, to the Aptimos, Calanus longitude, the same here in the table. And the tolerance of habitat components are smoothing parameters, and this N is the average neighborhood size that is corresponding to the amount of data used to fit individual points in the average number of data. So in this case, we see that delta smell increases abundance when you have higher biomass of Pseudoaptimos cynocalanus and intermediate levels of longitude, in this case, between the confluence and the lower San Joaquin and Sacramento rivers. For Longfin smelt, we see that the relative abundance during this baseline period increases towards the west, higher longitudes, also increases with Eurythemera, as shown below here, and Limnoitana as well. Then the pod years, the pelagic organism decline years, we see Cynocalanus is a species that is associated with larger abundance of delta smell, relative abundance, and intermediate abundance, Seriodaphnia, and temperatures. So in this graph, yeah, you see about around the peak between 19 and 21, the peak temperature for during this period. For Longfin smelt, during the pod period, we also see temperature as one of the primary factors. In this case, we have Limnoitana eurythemera that increase the abundance of Longfin smelt based on model output are associated with increase in abundance, and the opposite with temperature. Lower temperatures are associated with higher abundance of Longfin. And this model included 4 variables. So the 4 variables displayed here on the right graph, cladocerans, that added a little more explanation to the model, up to 58%. But that, compared to the previous variables, yeah, occur at the intermediate levels, the highest abundance of delta smelt. Now for the climate change intensification period, see a very different change. By this time, the delta smelt had dramatically declined. So you see the responses here are very different from previous ones, but still the response to Cynocalanus, the intermediate response to intermediate abundance of arpacticoids, and latitude, that's consistent with the remaining higher density presence in northern areas like North Delta, Caches Loop. For Longfin smelt, in the climate change intensification period, we still see Eurythemera as increase in Longfin smelt abundance and intermediate abundance of Longfin smelt for cladocerans, miscellaneous cladocerans, and low abundance for acanthocyclops. So in summary, looking at the previous models in 3D, there are two things that are worth pointing out. One is the similarity between the species, both in the baseline period shows longitude as one of the major explanatory variables, longitude, and in temperature in the pod period, both shows that. And the second thing is if you see the peaks of abundance, obviously there's over time, if you look from top to down, although it's log scale, there has been in both the species large decline. And in addition to that, the peak decline tends to be more restricted to particular range of the explanatory variables, unlike the earlier periods that there's higher abundance across longer range of explanatory variables. So in summary, we have abundance of both species increase with some zooplankton species. You see some of them in all the periods, like Cynocalanus for delta smelt, or Eurythemera in all the periods for Longfin smelt. Now the latitude and longitude, there are some gradients, strong gradients there. So from the confluence to lower San Joaquin, longitude is important for delta smelt in the baseline period, and latitude in the climate change intensification period is suggesting further habitat contraction in latitude. For longitude, we saw that earlier, baseline period towards the west, mostly abundance. And temperature, there are peaks for both species during the pod, around 21. This is based on a model for just for temperature. If we consider the entire response is lower than 21, the peak for delta smelt, temperature for Longfin smelt, the abundance will decline above or below this 15.5 degrees Celsius. So preliminary conclusions, despite a large reported shift in zooplankton biomass over time, some of the taxa appears to be important for both species. In the case of delta smelt, Cynocalanus, and Longfin Eurythemera, longitudinal and temperature gradients are for both smelt species during the baseline and the pod period, support habitat compression and further habitat compression for both smelt species during the climate change intensification period. So this is consistent with what we know from the decrease in the habitat quality and quantity over time for both species. So there's a need for informing the climate adaptation. So a lot of questions with implications for supplementation of species we'll have to deal with, whether it's based on some published studies there by UC Davis that shows that fish are not in good nutritional condition when they are recapture some delta smelt. So there definitely some need to also consider possibly need to augment the prey resources for this species. So the analysis of all these three different ways to analyze this data set show that, yeah, there's different explanatory variables that come into play. So every single conclusion needs to be addressed to particular questions for restoration or recovery of species. Since the prey, the importance of the prey will vary, but we have a climate change, and so I think I will leave it there in case there's some additional questions. Yes, Kim.
How do you think of habitat compression from just overall decrease in abundance leading to lower abundance?
There is in both species more I would call it, instead of using the same word, based on the 3D plots, more reduction in the area where the abundance is non-zero, and so it could be range from optimal that we saw in the baseline period, optimal to suboptimal to poor quality on habitat. So I think based on the modeling, it's clear that the range where the explanatory variables used to find delta smelt or Longfin smelt abundance, and they're now in the outputs. There are areas that no longer appear to sustain. I can go back to some of the slides. See, there's plenty in the last graphs. There's particularly more missing or near zero areas with abundance across this. If these are the major explanatory variables, then that brings the issue, yeah, that spatial representation will be useful, yes, in a map to show the analysis. But based on other studies, we know that there has been a decrease in the occurrence over time based on the recovery areas, with that requirement for recovery areas for delta smelt. They're less and less observed, although the hydrology, of course, influences when the fish will appear, but I think the difference in the habitat quality and concentration in smaller areas. Surprisingly, turbidity was not included here. That is very common, associated with delta smelt, but based on previous analysis, important variable depending on how we analyze intraseasonally. But yeah, so yeah, this is not the last outcome of the presentation. I plan to continue doing this with the collaborators and look for ways to make this of possible use for supplementation, analyze, predict values for the period that we didn't include beyond 2017, because we know that missing there it will be more difficult to model that particularly for delta smelt after 2017.
All right. Thank you very much, Gonzalo. Next up, we have Robert Fournier from Cal Berkeley.
Thank you very much, and good morning. I think it's still morning, yeah. Today, I'm going to be sharing with you some of the work that we've been doing regarding using phenology, in this case, kind of the timing of when populations reach peak abundance, to inform how we understand risk dynamics of fishes and their zooplankton prey throughout the San Francisco Bay estuary. And to do that, we first need to take a step back and set the stage. Of course, everyone in this room knows how dynamic estuaries are. They're transitional habitats, not just in the fact that they span abiotic gradients from saline to freshwater, but also in how many species use them. Fishes, for example, use estuarine systems in general for breeding and rearing grounds. And so that means they may be using kind of the habitat and the resources within the estuary and interact with different portions of the food web quite differently over the span of a relatively limited time. One of the things that we know about fishes in general is this age zero survivorship is often really, really important to how well the population can sustain itself. This is true across widely different taxonomic groups, and it's true for many of the species in the estuary today. However, the other side of this coin is that environmental threats are particularly impactful on age zero fishes. So things like habitat degradation or changes in flows or changes in food web dynamics might disproportionately affect these age classes, which then have cascading impacts up through the rest of the population. And we might have seen some of these dynamics unfold in the estuary before. As you know, during the pelagic organism decline, many pelagic fish species have shown dramatic periods of decline. While many of you in this room study some of the causes of the pelagic organism decline, things like entrainment or habitat degradation or limited food supplies, perhaps brought on by the invasion of the Asian clams in the 80s, which suppressed zooplankton stocks, which we found to be very important for many of these species, might be very important drivers of this decline. And that last kind of food web component is what we are interested in this study. How can we link predator decline risk with their prey to help us better understand risk dynamics in the system? Now, one of the most common tools that a conservation biologist might use to assess risk within a system is a population viability analysis. Many of these models seek to quantify the probability of a given species reaching a quasi-extinction, or we'll call it critical decline threshold, over a certain time horizon. And one of the big downsides, as we heard earlier on from one of our speakers, is that these PVA require extensive and very often rare demographic data, right? A lot of times we just don't know what is the age-specific survival or fecundity for many species, even those that are very widely studied. And so to overcome these and some other methodologies, time series-based PVA were developed that use time series of species abundances to assess the convergent properties of the system. They algorithmically search for kind of patterns in the noise and hope to estimate two primary parameters. The first is kind of error-free growth. Is the population going up or down? We call this U, but in the time series literature, you also might see this called lambda. And the second is process-based variation. So how much is the population varying over time as a result of environmental processes? And with these parameters, we can calculate risk much like you could with a more traditional PVA. However, one of the big downsides of these methodologies is that they assume risk is consistent throughout time. And for age zero fish species in an estuary, this might not hold, right? They're using different resources, they're interacting with different food webs, even within 1 year. So assuming that risk is going to be the same across that year might not hold. And so what we wanted to do with our study is to use our understanding of some of the phenological dynamics of this system to better construct PVA and assess risk throughout the estuary. And so using data from the CDFW base study and the IEP's environmental monitoring program for age zero fishes and zooplankton respectively, we took those time series, which as you know are sampled monthly, and broke them into their constituent months. So we created a yearly time series of all the Januaries, all the Februaries, all the Marches, and so on and so forth. We used multivariate autoregressive models to estimate those growth and process error parameters. And we did so by feeding all of the data from the sampling station in each region to inform kind of one regional growth and variance estimate. And with these, we calculated the probability that a species in a given region at a given month would experience a 90% decline. And we're going to call that the critical decline risk. And we also assessed risk across what we determined to be important phenological windows. So using, again, historical abundance trends to predict when risk might be most acute. and then the estuary for fishes and their prey. And we'll talk a little bit more about how we did that later on. Now, our goals with these models were, first and foremost, just to get a general sense of what's going on with these populations. So see if and how population parameters, including growth and variance, varied across species and across time. We assessed if the risk showed differential patterns throughout the year, so if months were different from one another. And we also wanted to look in particular if low abundance windows were different than high abundance windows. And then finally, we wanted to bring in the trophic dynamics of the system. So we compared risk between predators and their prey or potential prey. And moving on to results, like I said, the first thing we did was look at just a general sense of the population here. So to orient you to this graph, each dot kind of represents the species, month, region, stratum that we were talking about. And they're grouped by colors into different species, and we fit ellipses to them to better visualize the data. The x-axis is this growth rate. So if you're to the right of 0, you are growing. And if you're to the left of 0, your population is declining. On the y-axis, we have process error variance. So the further you are from 0, the more variable your population are. And one of the things that we can see is that there's lots and lots of negative growth rates, unfortunately. And that's not surprising to many of us. In fact, we have some species like our good friend, the Longfin Smelt, here shown in this lilac-y color that shows nearly universal decline trajectories. But one of the things that's true about them is they're doing it pretty consistently. Like they're declining without a ton of variance, which is not necessarily great for them. On the other side of things, we do have some species that are doing okay on the growth side of things. Like this is the northern anchovy, and it has lots of positive growth rates, but it's doing so in a highly variable way. And one of the things that's true about how we calculate critical decline risk is it's not just the trend, but it's also how much you vary. So obviously the longfin smelt might show high critical decline risk because it's declining, whereas the northern anchovy, just by a basis of it being really, really fluctuating, right? Just through random chance, it might blip out in any one of these months. So that can show like similar patterns of decline risk. And so that's what we did. We used these population parameters to assess risk for each month of the year. And again, to orient you to this graph, the x-axes here are the different months. Each kind of facet is a species. The colors represent regions, which we will use throughout the rest of the talk. Highly saturated colors indicate that these are high abundance periods, but you'll also notice a lot of gaps. These are when the time series contained more than 70% zeros, because assessing the risk of a species dipping below 0 is kind of not necessarily all that important for us because you can't really dip below 0. And one of the things that we can just see with our kind of eyes is that risk is not consistent across the year. You can see dips and valleys. And so this assumption that risk might be kind of uniform across the entire first year of their life seems to not hold. But one of the things that we did test is, again, these high abundance versus low abundance windows. Does having more individuals within a region at a given month kind of buffer you against critical decline risk? And we found that it doesn't. One of the reasons that we think this might occur is because if the system is food limited, right? If these age 0 individuals are having trouble finding their zooplankton prey, density dependent factors, when there are a lot of species or a lot of individuals within the region, might be bolstering rates of risk, even when compared to periods of low abundance, which you might expect be more likely to blip out. Okay, so now we have kind of risk estimates for each species, month, region, stratum, and we wanted to kind of bring that back to phenology. And so we assessed what we're calling phenological windows that are based on historical abundance patterns. So we have a fish here, right? And we calculated what period of time contains 80% of the expected catch for that year. And right, we determined that to be the important phenological window, but we also wanted to do this and look at their prey items. So we also applied this window to the suite of zooplankton prey that that fish might encounter, regardless of what that zooplankton is kind of doing within that window. So we're only interested in the zooplankton in the period of time that is important to the fish. And when we do that, we get something like this. Again, colors are going to represent the region. On the left-hand side in the solid lines, we have the fish species, and then on the right-hand side in the dashed line, we have zooplankton. And the range represents the difference between kind of the best case risk scenario and the worst case risk scenario. So in essence, it's error bands. And when we apply this to all of our species region strata, we get a crazy graph that we could probably spend all day looking at, but because this is the session right before lunch, I will not do that to you. We did see some general patterns. First, while individual fishes might miss individual zooplankton taxa, that might not be all that important because many of the fish species that we're including are pretty generalist. They're going to eat the zooplankton that they come across. And while like individual taxa might be important, in general, we can assume that they're going to be relatively generalist. But when you compare fishes against the whole suite of their prey, so while they might miss individual prey items, they are very unlikely to miss the whole of their assemblage. And so this is at kind of the current time point. And that kind of makes sense, right? We're not going out to the bay and seeing these large scale decoupling events happening today. But what we wanted to do is then project that risk into the future. How might this look over the next 10 years? And when we do that, we get something like this. So the x-axis here is basically years from today. The dashed lines are still zooplankton, the solid lines are still fishes, and the bands again represent the difference between best case and worst case scenario. And again, applied to all species, it looks a little something like this. We'll notice that a good chunk of those, the fishes and zooplankton, stay kind of synchronous over time. But for a not insubstantial subset, we see that fish and zooplankton risk begins to diverge across the projection. Now you'll notice a lot of pink, which is in San Pablo Bay. And in all of these circumstances, the rate of risk for fishes is higher than that for their zooplankton prey. So the fishes are kind of flashier and more unstable and have a higher risk of experiencing a critical decline in their zooplankton prey. But there is one interesting counterexample, which is the striped bass in both the delta and the confluence. These species are comparatively more stable than the rest of the fishes that we've modeled. And in fact, their zooplankton prey are more at risk of experiencing a decline than the striped bass are. We think, again, this might have to do with things like the Asian clam, which is more acutely suppressing zooplankton in the fresher parts of the estuary, whereas when you pair that with this comparatively stable striped bass, we see these dynamics unfold. But this situation is of particular concern because if fishes get to a place and time where there are no zooplankton, that obviously collapses the food web anyway. And so next we wanted to kind of scale this to the whole of the estuary up top, or at least the whole of our modeled regions of the estuary up top, and then each subregion in the bottom. So these are all fishes and all zooplankton across our models. The first thing we can see is that zooplankton, again in dashed line, this time in the green, have much wider ranges of uncertainty than their fishes. Now, this is a little bit intuitive in the sense that we have many more zooplankton taxa in our models than we do fishes. So there's lots of different pathways that can be incorporated. But zooplankton are also they turn over much more quickly. There can be more boom and bust. So the uncertainty regarding zooplankton projections into the future grows much faster than fishes do. Another pattern that we see is kind of hinted at from earlier. One of the strongest areas of potential divergence we see occurs in San Pablo Bay. This might have biotic and abiotic components. On the biotic side of things, fishes that kind of inhabit this region more continually, like that are more marine associated, like the northern anchovy, as we discussed earlier, are just very variable, and that's increasing kind of risk throughout the region. If you have kind of a more freshwater associated species that may use San Pablo Bay for a limited portion of its lifetime, again, that kind of limited use might also be reflected in our risk estimates. And on the abiotic side of things, as you all know, San Pablo Bay is kind of the saltiest region that the EMP and Bay study overlap. And so you not only have kind of the influence of the freshwater dynamics that you might see in the inland parts of the estuary, but also much more of a marine influence. And this kind of double whammy bookend of things, like you may be seeing influences of marine phenomena like marine heat waves and things like that, that might be more acutely felt in this region. So to kind of bring this together, the first thing that we saw is that many fishes are not doing so great throughout the estuary. There's varied growth trajectories and process-based fluctuations, but negative trends were common. In fact, that's the most common trend that we saw. And the positive trends that we saw were also highly variable. We saw that risk varies a lot throughout the first year of life. So this assumption that if we are assessing risk at that scale, you might be consistent, might be over or underestimating risk depending on kind of which species and region you're looking at. So if we want to kind of manage and conserve these species, we need to have a more granular understanding of when and where they are at risk. We also saw that high abundance months are not safer than low abundance months. And again, density dependent factors within the estuary might be contributing to this. We saw that in the present, the risk for predators and prey was pretty similar. Even though individual fish species might miss individual zooplankton taxa, for the most part, you're not going to miss the entirety of your assemblage today. However, as we project this risk out into the future, we see a lot of decoupling between predator and prey assemblages. But of course, zooplankton have a much wider range of uncertainty associated with them, and so we need to take that a little bit with a grain of salt. We also saw that for the most part, when we saw divergence, it was generally happening in San Pablo Bay with kind of one species as a counterexample. And so that region is of particular interest if we are looking at the probability of experience, this trophic mismatch hypothesis, right? Predators missing their prey. And to kind of zoom out to the big picture, in 2022, the United Nations put out a report that identified phenological change and this idea of trophic mismatches as particularly important issues of environmental concern. So as we seek to manage and conserve these fishes, understanding that their phenological dynamics might change and understanding that those phenological dynamics can then lead to changes in their trophic status could be really important for our understanding of the estuary as a whole. So first, I want to say thank you to our funding. We were funded by the CDFW Prop 1 grant. And then, of course, you can't not thank the many field staff across all of the monitoring programs that have collected these data for the past 45 years. And that couldn't do the study without. With that, I'm happy to take any questions. Over. Okay. All right. One question, I think. Yeah, Wim.
I like that you bring up the point about density dependence, and I've been thinking and working about that on that for a while, sort of on the side, trying to figure out how that comes into play with our analysis. But it's not clear to me at all that that's a competition among different groups of zooplankton or within zooplankton species.
Right.
Because they live in a moving, diffusive framework.
Right.
And so you can't say that this bug will be food supply or that will there.
Right.
Because over the time frame, the time that they're in that synthetic water mass, there's just good water.
Right. Yeah, and I think I should maybe caveat that saying we didn't explicitly test density dependence or any competitive kind of structures amongst either the different prey items or between predators themselves. We were just kind of looking at that as a potential way to explain it. And I think one of the things that I would love to do is look at more competitive interactions. I'm doing that with some of my future work, like looking at kind of how kind of the density dependent factors of fishes affect kind of population dynamics into the future, but that was not part of this work. Yeah. Right. So, but thank you. Yeah, we don't, I unfortunately could not test it with this work, but I think that future studies should definitely try and incorporate that with more fine level detail. Okay, thank you all.
Thank you, Robert. All right, next we have Jorge Arroyo-Esquivel with CDFW.
Hi. Thank you, everyone. So I'm going to talk about how we identified a possible relationship between white sturgeon population dynamics and hydrology. So today I'm going to talk about some extensions of Blackburn model, which is the most recent life cycle model that has been developed for white sturgeon in the estuary. The process that we went through to create extensions to the model. And then thanks to the previous speaker for introducing PBA. So I'm going to talk about PBAs as well in this presentation. So let's get started by asking the question of what are the possible extensions that the Blackburn model can. Let's start with the model itself, right? This model separates sturgeon females into 21 age classes and looks at the population year over year. At any given year, a proportion of sturgeon at each age class experience mortality. And the ones that survive to the next year, they move up to 1 year age class, except that those that are in the age class 20 are going to be assumed to remain in that age class until they die. Also, starting at age 10, the females will start sexual maturation and they will start providing new yoys into the population. And the model assumes that the fecundity increases year over year with each age class, and it reaches its maximum at age 20. That's why we're only keeping track of 21 year class instead of like 50, right? So this is the base model that was introduced in 2019. And we were thinking about how this model can be extended, and we identified 3 opportunities for it. That could be either creating improvements into the dynamics, making them more described by data, or deepening the analysis that can be done to the model. So let's start with talking about stochastic recruitment. The model originally assumes that recruitment year over year is completely random. Basically, sturgeon come and they roll a die, and if it lands on a 6, then recruitment happens that year. So we worked on identifying how this stochasticity could be described using hydrological conditions in the delta instead of pure chance. We also incorporated the risks of hub mortality in all age classes, given the new information that we have from water years 2022 and 2021, that where sturgeon face a lot of mortality due to harmful algal blooms. And also the model can be analyzed by, further analyzed by exploring population dynamics over a longer time span. And I will talk about that a little later in the presentation. So let's get started and talk about how can we address these extensions. Let's start by talking about how to identify a possible relationship between hydrology and recruitment. We identified an outflow threshold for successful recruitment by linking the catch of yoy white sturgeon in 20 millimeter and base study surveys with delta outflow from day flow. We created general additive models or GAMs under several aggregations of winter and spring flows of outflow to identify which one is the best aggregation to predict the likelihood of yoy catch in a given water year. And our analysis found that the aggregations of January to May and February to March are the models that better predict the yoy catch in a water year. And throughout the rest of the presentation, I'm going to present results only using the February-March aggregation. We identified that these 2 models are not statistically significantly better one over the other. So just for the sake of simplicity, I'm just going to present about February and March aggregations. So this is the output of the general additive model. And we observed that 90% of the water years that were classified as above normal or wet had at least a catch of 2 yoy white sturgeon. And we identified that expected catch of 2 is significantly smaller than, well, the expected catch of the model output is significantly smaller than, higher than 2 after 25,000 CFS. So we identified 25,000 CFS as this threshold for to determine whether we're going to have successful recruitment or not in the model in a given water year. And when we look at the actual data since 1997, all wet water years and all but one above normal water years had a mean daily outflow in February and March higher than 25,000 CFS. So that is pointing towards the fact that this threshold makes sense in the context of hydrology. And this allows us to expand the model by incorporating outflow as a factor that determines recruitment. And we will say that recruitment will be successful at any year where the outflow is during February and March averages above 25,000 CFS. We also include hub mortality by taking a random probability that there will be an intense hub event at any given year. There is no evidence to support an increased mortality with different age classes of not only of sturgeon, but we couldn't find any evidence of other fishes that had different mortalities at different age classes. So we're just going to assume that hubs affect the mortality of all age classes with an equal intensity. And now let's talk about let's talk about uncertainty of growth rate. This model is, at the end of the day, still stochastic in nature. So we want to analyze the growth rate of the population by basically running it and calculating the growth rate using the geometric mean of proportions year over year. And this is an illustration of how this uncertainty in doing this estimate decreases as we increase the number of years that we let the simulation run. I'm only displaying 20 to 25 right now because that's the time span that was used in the original model. And we identified that by letting the model run for longer, and now that we actually have estimates of outflow that are driving recruitment, we can see that these variance decreases significantly as we increase the number of years used in the simulations. And that is pointing towards having better estimates of what will happen to white sturgeon in the long run as we increase the number of years that are used in the simulations. Okay, now let's talk about population viability analysis on this new model. And we're gonna ask a simple question. Is the white sturgeon population in the Delta viable? Just gonna quickly talk about what I mean by viability here. We're gonna be looking at a parameter lambda, which is the estimated long-term growth rate of the population. And we're gonna say that a population will be viable when this lambda is bigger than 1. Basically, what you're expecting in this toy example is that when lambda is bigger than 1, the population will increase year over year. Whereas if the population is not viable, that's when lambda is smaller than 1, and you'll see a year over year decline in the population. So we use our new model that incorporates outflow, incorporates hub mortality to estimate the white sturgeon viability, varying the parameter values of the H0 survival probability and the H3 plus survival probability. So in this model, we're assuming that the mean survival of all H classes levels out at H3. So basically all sturgeon H3 plus have more or less the same survival probability. And the heat map looks something like this. Doesn't tell you much, but let's put some more lines on it. This white line represents where the growth rate equals 1. So everything above this white line represents a viable population, and everything underneath represents a non-viable population. So this gray line, it constitutes the observed growth rate that was estimated using abundance estimates provided by fisheries in CDFW. That shows that since 1979, there has been estimated a decline of 3.5% year over year in population abundance. And that will, in the context that I just presented, that will put the population of white sturgeon as not viable in the current conditions. And if we use the baseline parameter values for survival probability of H0 and H3, we can see that our estimate of the estimated growth rate is pretty close to actually observed growth rate using these abundance estimates. And that is telling us that the model in some ways is following what the dynamics should look like. Here I'm going to present a global sensitivity analysis that allows us to identify which parameters of the different parameters that we incorporate into the model are going to be the most important. We're sorting them from most important on the top to least important. And as you can see, the H0 and H3 plus were identified to be the most important parameters. And it's not too close. The H0, the mean survival probability of H0 sturgeon is miles ahead of the rest of the parameters in this analysis. And that is telling us a lot of the importance of these younger age classes. And then quickly, I'm going to talk about can improve outflow and juvenile supplementation provide a better future for sturgeon. We see that right now the future is a little bleak. So what can we do about it? And we can look at, for example, trying to do improvements in outflow conditions. Basically, if there's a world in which outflow conditions can be improved to have a more successful recruitment year over year. Right now, there has been estimates from anywhere from 4 to 8 years between successful recruitment events. And you can see that as we increase this frequency, hopefully through improved outflow conditions, we can get the probability of having a viable population reach around 25%. And furthermore, if we overcome the survival of Yoy's white sturgeon and introduce H1 white sturgeon through supplementation, we can get this probability of a viable population even further. Somewhere like we can reach 50% probability of viability around 16,000 white sturgeon supplemented based on the numbers estimated by the model. So in conclusion, we extended the Blackboard model to include more variables of interest and also with the information that we have gathered about white sturgeon dynamics from the past 5 years. And we could see right now we are at around 10 to 15% probability of viable population, but we could improve it even further to up to 25%. So that's a 10% increase if outflow conditions are improved in the delta. And for supplementation, there's a potential for an increase of beyond 50%. And besides the estimates and the predictions that we're trying to make with this model is also highlighting the needs that we have to understand certain factors of white sturgeon dynamics. For example, fecundity. We know that recruitment is successful. It's more likely to be successful the wetter the year is, but that doesn't tell us whether during drier years white sturgeon are actually spawning or not, and that could guide us into finding other ways to make a more likely successful recruitment in a drier year. And also yoy survival. The estimates for yoy survival that we have for white sturgeon are actually based on lake sturgeon, which has a completely different life cycle. So it's surprising that we got such a good estimate with lake sturgeon data. So it will be great if we can actually get better estimates for white sturgeon in the Delta. Yeah, thank you. I will take any questions.
Wait for the mic too. Thanks.
Well, I just wanted to ask, what would be the mechanism for a HAB effect on sturgeon?
I'm not entirely sure. It could probably be sublethal effects caused by decreased oxygen. It could also be toxicity. It will depend a lot on what types of what the main algae that's produced in the harmful algal bloom will be. Different fish have different responses to different algae, so there could be several factors. Yeah,
Sturgeon look like just such tough animals.
They are, but there were huge estimates of decline. I don't have the numbers right now on top of my head. During the 2021 harmful algal bloom that occurred in the Delta. So yeah, it will be like sturgeon are tough, but it seems that harmful algal blooms can have a big potential to harm them more. So yeah.
Thanks, Jorge. Great talk. My question is about, and maybe you don't know, but do we know where the 8-0 fishes are occupying? Where do they go? What are the important habitats for them?
I think they go pretty fast from at least what we know from the Sacramento River. There's still a lot of unknowns of what's going on in the San Joaquin. But from the Sacramento, they spawn somewhere around between Knights Landing and Colusa. And they are quickly taken down downstream somewhere in they slowly get a higher salinity tolerance, and then they slowly start spreading throughout the Delta.
Thank you.
Hi, great presentation. I had a question about the GAM that you did of the relationship of sturgeon with hydrology. It looked like there was initially a really strong positive relationship. It went up, and then as outflow further increased, the population went down.
Yes.
And then the subsequent slide showed it seemed to be just one data point from a below normal year that was at the peak of that curve.
Yes.
Yeah, so I'm just wondering whether you might want to consider fitting a different model that might not be so influenced by like one outlier point, or if you think that one point is a real phenomenon.
That is a great point. Thank you for pointing that out. And yes, this huge bump at the like around 25,000 CFS, it's driven a lot, like it's highly driven by this outlier, which is water year 2017. So it's an option to explore, like to try to maybe take out that point in particular, so we can have like a smoother line without outliers. We decided to keep the outlier But if you go to the global sensitivity analysis, where is it? Here, you can see that the value of the outflow threshold didn't end up being as important. We tested different outflow thresholds from 10,000 to 40,000 CFS, and it was not as important as of a driver, like the numerical value that we were inputting into the model, compared to yoy survival, which is the biggest unknown that we have at this point.
Okay, yeah, that makes sense. I guess something to think about is just whether there's a reasonable mechanism behind an initial increase in outflow as flow increases, and then a huge decrease, and then followed by another.
I think that decrease is mainly driven by the outlier. If the outlier, I will have to run it, but I would imagine that if we take out the outlier, we'll just see a linear increase. Maybe not linear, maybe just some bumps, but linear in general.
Okay, thank you.
Sure. Thank you.
Thank you, Jorge. Next up, we have Delia Carpenter with Scripps.
Hi, today I'm presenting on recovering high-frequency plankton dynamics from low-frequency samples. The Sacramento Delta has a rich data set of physical and biological variables, which has produced valuable insights into the ecosystem structure and function. However, important ecological dynamics remain hidden because plankton dynamics occur on timescales that are impractical to sample. For instance, monthly sampling of phytoplankton. Wait, I think this might be the wrong version. Was there two versions on there? Sorry. Are you sure this is the right version? Okay, can I try again? Sorry. Okay, sorry. Can you see that? Okay, sorry. Okay. So, yeah, as I said, the San Francisco Bay Delta has a lot of data, but key ecological dynamics remain hidden. Plankton form the foundation of the ecosystem and are crucial for understanding higher trophic levels, but their population dynamics unfold on timescales that would be impractical to sample. As an analogy, sampling phytoplankton every month would be like sampling trees every 400 years. Modern scientific instruments now provide near continuous monitoring of environmental conditions, which offers new opportunities to uncover these undersampled plankton dynamics. But integrating these two data sources can be challenging, and it's often conceded that data on the coarsest scale will dictate the resolution of the analysis. Empirical dynamic modeling offers a potential solution. EDM is a time series analysis method that learns patterns from time series data using state space reconstruction. To explain this, I'll go through the figure on the right. It shows 3 time series, X, Y, and Z. It's hard to say from just looking at the 3 time series what the relationship between these variables are. But if instead of plotting the data as 3 separate time series, we plot it as a moving trajectory in a 3-dimensional coordinate system, a clear picture of the system dynamics starts to emerge. This picture is called the attractor, and it helps us to understand how the variables in the system interact with each other. For example, notice how in the left half of the attractor, X and Z are negatively correlated, while in the right half, they're positively correlated. EDM can pick up on complex patterns like this, where relationships between variables are not constant, but change over time depending on the system state. These changing relationships are a hallmark of complex systems. The most important thing about EDM and what makes it unique in ecosystem dynamics modeling is that we can reconstruct this picture or model of the system dynamics directly from the data without knowing the explicit equations that govern the system. If we were just given the time series of X, Y, and Z, we could figure out how they interact with each other directly from the data without any preconceptions. This makes the method ideal for studying complex systems where we don't know the underlying equations. The main assumption of EDM is that the system follows deterministic rules rather than being purely random. This assumption can be validated using out-of-sample prediction. Let's see how EDM can reconstruct the value of plankton that wasn't observed by identifying patterns in the data. Suppose we have a time series for wind speed and temperature, which influence plankton abundance. This is just a made-up example. However, let's say plankton wasn't observed at time 50. Can we estimate its value using EDM? So the first step is that we find times that had similar environmental conditions. Here we see that at time 50, there was low temperature and high wind speed. In this example, we can just look for the similar environmental conditions by eye, but we can also do it by plotting those environmental time series in a state space and identifying nearby points. So here we see this red dot representing the state of wind speed and temperature at time 50, and we find just the nearby points. So in this case, we see that times 32 and 86 had the most similar environmental conditions. Translating this over to the time series on the right, we see that we have identified those similar environmental conditions with the low temperature and high wind speed. So to reconstruct the missing plankton value, we simply average the plankton observations at those times that had similar environmental conditions. So if you compare the yellow prediction with the actual data point that was removed, you can see that the prediction's pretty accurate. So notice that the plankton doesn't need to be sampled frequently or evenly to do this, and that we can reconstruct the value of plankton at any time point where we have observed the environmental variables. This means that we can reconstruct a high frequency plankton time series, even if the plankton samples are infrequent or uneven. So here's an example with some real data. I applied EDM to data of northern anchovy larvae from the Calcofi survey, which is one of the longest running ecological monitoring programs. I also used daily environmental data from satellites. In the figures below, you can see the vastly different sampling resolutions. The fish larvae are sampled on an inconsistent seasonal basis. They're the pink dots, and the environmental data is sampled daily. It's the really high frequency data in the background. Calcofi data has mostly been used to understand long-term patterns in fish larval abundance and distribution because of this uneven sampling on finer timescales. But I will show how with EDM you can learn about the high frequency dynamics even from seasonal samples. First, I looked for correlations of the environmental variables with the larval observations. I tested sea surface temperature, wind speed, and chlorophyll, averaging them over daily to seasonal timescales, and none of them had a strong linear correlation with the larvae observations. The highest was around 0.15. But an EDM model reconstructs the larval observations with a correlation of 0.53. This is a pretty good result because these are individual observations that are probably pretty noisy. By contrast, a multiple linear regression model using these same predictors achieves a correlation of 0.17, which is not as good. Remarkably, EDM reveals daily to weekly dynamics of anchovy larvae, even though they were only sampled seasonally. Using these environmental drivers, you could reconstruct a high resolution time series of anchovy larvae across the California current ecosystem. This example shows how EDM can integrate data sets with vastly different resolutions to uncover undersampled plankton dynamics. In conclusion, EDM is an emerging tool that can enhance the value of long-term biological monitoring programs when paired with continuous environmental data. By uncovering biological dynamics on timescales that would be impractical to observe directly, EDM provides new insights into ecosystem processes. EDM can be used to reconstruct high-frequency plankton dynamics from low-frequency samples. Another potential application might be to extend plankton time series backwards into historical periods where they weren't observed, even when environmental data is present. EDM could be a powerful tool for integrating diverse data sets across the Delta, allowing us to get a more complete picture of the ecosystem dynamics. So thanks, and this is just a link to a paper that's kind of similar to this project. So yeah. Yeah.
Thanks, that was really cool. I'm just wondering, when you're going about selecting which time series you want to put into one of these scenarios, how do you go about selecting them? Like, if you pick any environmental time series at random, it's not necessarily going to have a relationship to the response.
Yeah, so you do that in a similar way to testing other models, where we do like a leave one out thing, where you remove a data point and you choose a model, and then you like remove one data point at a time and try to predict that value. And then you just see how well did that model predict the withheld data. And then that means it was a good model if it does well at that.
That was really cool. Thanks, Delia. I was wondering, do you think that this type of model could be used similar to the work that was done yesterday, looking at zooplankton, kind of how the monitoring programs are organized and developed in terms of, certain areas are oversampled, trying to make those kind of determinations? Do you think that you could use this model to get that kind of information?
Yeah, I think you could definitely do that. Since like you can use EDM to reconstruct values that weren't observed, you can kind of see, will a survey at this spatial location be able to reconstruct this other one? Like, for example, are they kind of redundant? Does one contain information about the other enough to reconstruct it? So you can kind of use that to figure out what sampling frequency you really need to get all the information.
Great. Thanks for the presentation. So sort of two questions combined. One is, does this work when the system is non-stationary? And the second is, how much data do you need to have in order to see a pattern?
Yeah, that's a great question. It can actually work when the system is non-stationary, like for example, when there's been a regime shift, but you just have to have enough data in the new regime that you can learn those patterns. And in terms of how many data points do you need, that depends on like sort of the sampling frequency, how complicated the dynamics are. Sometimes it's suggested you need around 30 data points, but it really depends on like the generation time of the species you're interested in and everything like that. So you just want to observe enough data points that you've maybe seen like enough generations of the species that there's some patterns emerging.
You used in your examples to demonstrate the point, you used just 2 or 3 time series and combine them. But I'm curious, are there any limitations on the number of time series? Are there any minimums or maximums that you can envisage?
You can usually test, you can test a lot of time series. Like you could make as many dimensional model as you want, but it usually turns out that like models over 10 dimensions are kind of don't work as well. Like, so you kind of test different models and it usually ends up not being that high dimensional.
All right, thanks, everybody. So we've been made aware of some safety incidences recently. So as a reminder, just be aware of your surroundings when you head out for lunch. Keep an eye out for cars. We suggest traveling in groups. Just be aware. All right, and we will meet back. You know what time we meet back? 1:50. Thank you.
Session 9, environmental stressors on salmon and smelt. I'm Florian Mauduit and I will moderate this session. So we will have 4 full talks and 3 lightning talks. And to begin with, we have Brett Harvey from CDFW. What do I touch? So use the keyboard arrows to go through your slides and read the scrolling numbers. Okay.
Hello, everybody. Welcome back from lunch. I hope you can hear me. I ran back here because I got turned around, I spaced out and walked toward the highway. So that tells you the state of mind I'm in these days. So I'm going to talk a little bit today, about 15 minutes, maybe 20, about a synthesis effort I've been working on with some colleagues of mine. It started a little, almost a year ago. And really, they're listed on the slide in no particular order, randomized. But what prompted this synthesis was that there's just a lot of new information about salmon that's come forward in the last couple of decades, but really even in the last several years or so. Plus we have the impending climate change. We have really interesting new restoration efforts, like you can see there's like a big juvenile capture thing up there somewhere. Efforts to reintroduce salmon above dams. We have new collaborations going on, and as symbolized by the slide down at the bottom, particularly with the tribes in many of these endeavors. And just really, just thinking about spring run now, they're really headed really quickly the way of winter run, in particular some of our more sensitive and important populations. So I just want to give a little kind of a disclaimer that what the synthesis is. It's really more of a thought piece. It's become more of a thought piece. And I looked up some of these words on the internet because I didn't know them, but we're trying to come up with like a contemporary episteme, or is it episteme, which is basically just a current scientific knowledge around salmon, but also what is the scientific zeitgeist? What's the spirit of the times about thinking about salmon and salmon management? And we also plumbed back and looked at pre-European ecology and reconstructions that to sort of provide a picture for where we might go and hopefully come up with some kind of a future vision. And this is still a work in progress, hopefully coming to a close soon. But I also want to say what this is not, is it's not a new salmon strategy or plan, nor is it a critique of existing plans. Instead, is really just a borrowing from many of these plans, colleagues, ideas, and just our experiences to think about salmon and where we're going with management. So what we did is like I said, we thought about what was. And really, we still have, though in a much reduced state, one of the most diverse groups, or the most diverse group of Chinook salmon across the global range. And the reason why is, I'm going to read this on the slide. We have a very diverse landscape and climate. Salmon have a highly migratory behavior, but they also have this ability to both retain behaviors that match their particular environment, the population's environment, but also have a lot of flexibility within that so they can respond to change. And so I put together this sort of bunch of cartoons to really just kind of show this broad picture of the diversity of salmon. And I think most people are aware of it, but you had adults either migrating, holding, or spawning at pretty much any given time of the year, sometimes all three in a single season. Had a lot of habitat and access to cold water mountain streams, which promoted a yearly life history, which is particularly important life history because it, especially now, I'll talk a little bit about that under what is. And you also had these winter pulse flows that would disperse young of the year salmon throughout the Central Valley floor, provide lateral connectivity to floodplains, longitudinal connectivity to the estuary. So we essentially had salmon rearing all over the place in the Central Valley watershed. So one thing I left out of this picture for now, but it's really important, was that there was also a salmon people, and these were the tribes. And these are tribes that co-evolved with salmon. And because of that, they developed a stewardship that was based on reciprocity. They took care of each other. And essentially, if the salmon died, the salmon people would die. That's the way I put it. But a really particular aspect of this was that harvest was managed at the tributary scale. And they ensured that every year enough adults made it upstream to spawn and fill the habitat with juveniles. And that was really important for maintaining populations and that spatial and life history diversity. And I was going to say that there wasn't any ocean harvest at that point, but I forget, there were killer whales out there. So contrast that with what is. And we kind of go across. I was going to do these one at a time, but I feel like I'm going to run out of time, so I'm just going to splash them all up there. The first thing you notice is, obviously we have dams, right? What do dams do? They block access to almost all of that historical cold stream habitat that was up in the mountains. And that really has made the yearling life history just almost like a ghost in the system. And that's really critical right now with climate change. And you may have noticed that the sun at the upper left got a little bigger and a little meaner. Because what we're finding is that yearling life history is the most resilient life history to drought. They can over summer up in those cold mountain streams, and when a lot of the salmon on the valley floor would succumb to the heat. We've also lost, because of the dams, these flows. We had winter pulse flows that we don't have them anymore, really, that would the ones that disperse salmon into the estuary. And thinking about climate change, that estuarine life history could be particularly important going into the future, because those salmon are downstream of what you'll see over on the right in the spring and the summer, is the establishment of a temperature predation gauntlet that's happening earlier and earlier on average every year, and essentially just causes the survival of out migrating smolts in the spring to plummet. But the salmon in the estuary are already past that. And you could probably plug the North Delta into that too. We've also lost lateral connectivity to floodplains, so they no longer flood as frequently, and salmon can't really access them. And then the other thing you might notice is that there are kind of these weird little grids that are downstream of the reservoirs are hatcheries. So we have what in their recent book called Managing Extinction, which I rushed through before this talk by Williams and Lichitoge, where they really lay out a case how the hatchery commodity system, where we manage salmon as a commodity, has really supported not only the issues that we have with the fitness of the natural populations due to introgression of fish that haven't really been selected for the natural environment, but also supports a mixed stock fishery. And the more hatchery fish you have out there in the ocean in proportion to natural fish, where we can't actually tell with many of those fish which ones they are, and then you provide those fish for harvest, that impacts the natural populations. Of course, we've had those closed down now for several years. But just a whole tangle of issues that are going on. So really, we start thinking about this whole thing in terms of the 4 H's, right? Everyone thinks about oops, I gave that away. Everyone thinks about the 4 H's. You have harvest, hydro, which in our case is more dams, because hydro is more of a Pacific Northwest Columbia River issue. We have issues of reduced habitat, and then of course hatcheries. But, we felt really constrained by that. And we thought, we thought of 2 more H's. We have heat, right, with climate change, which is becoming a more exacerbating issue with all the other things going on with salmon. And then we also have this human-salmon connection, or maybe the lack of it, but we know that that's important, that human-salmon connection. And then we realized that, okay, we're still something wrong here. And we realized that, well, this is kind of like an un-H, that the way that we approach salmon management is sort of like an unholism. Don't call someone that on the street. But a little bit more on this unholism idea, which is essentially like reductionism. And it's really, let me go back a little bit. It's a really issue, not like a wicked problem like we talk about, but really an issue of the Western scientific engineering mindset, where we don't really think about things as full systems as frequently as, and then maybe it's a human thing that we try and divide to tackle problems, but we end up dividing and not conquering. And because we're just looking at little like pieces and addressing pieces. And this sort of leads to these things that, are called fixes that fail, where you're addressing these short-term issues. And these actually can often end up leading to more negative consequences at larger ecological or conceptual scales. And so I just want to challenge everybody to think about the work they do and what sort of components might fall into that, because it's pretty much most of what I do every day. So a little bit more on holism. The idea is that the whole is greater than the sum of the parts. Parts of a whole are in intimate interconnection and cannot exist independently of the whole and cannot be understood without reference to the whole. So this is something that we're like, oh yeah, we're really onto something here. And in the context of salmon, this really just thinks about whenever you're thinking about a salmon management action or a strategy, about thinking about every action within the full context of the salmon's life cycle across the land and oceanscape. Super easy. So we came up with these 3 guiding principles. And again, this is where it gets, we step back a little bit. And the number 1 principle is really to promote spatial SLD, spatial and life history diversity. Through expansion of diverse and connected freshwater and estuarine habitats, but really this means expanding opportunities for salmon. That's what we need to do, is really focus on expanding the opportunities and let the salmon do their thing. So this might mean access above dams, and this is where I'm borrowing from a lot of these existing frameworks. Increasing flood frequency in flood plains or access to flood plains, increasing dispersal into estuary. And a lot of this happens, through functional flows. Sort of the flip side of that is to exercise discretion on actions that are just focused on preserving like a really distinct ecotype. And so you think, oh, the Smelt ecotype, or and I talked a little bit like the yearling ecotype, but really trying to like engineer a particular ecotype and then really focus on that to produce to like increase abundance. Maybe not the best long-term approach. And again, you really just want to promote the opportunity diversity. I'll be saying that a lot. That gives rise to diverse ecotypes. And then finally is this aspect of supporting a thriving human salmon connection. And this is kind of going to be a challenge, right? Because really that happens through harvest and consumption. That's how we interact with salmon. And that might be difficult with our huge population size to produce I mean, it's a challenge. It's going to take education awareness, making salmon a part of the community fabric, all these sort of things. And salmon has a sense of place. But, you think about all these things together and like these ideas. And again, this is borrowing from existing strategies, but what could be? Or maybe we should be like all like aspirational, what will be? But you can see maybe toward the top and around the dams, this idea of reintroducing salmon above dams and access essentially to habitat opportunities above dams through trucking up and down. And that's people are actively working on that right now. And the Big Notch project that there was a talk on that yesterday, where we increased both the flood frequency and access. There's potentially winter pulse flows to try and move salmon around the system a little bit. Then we're going to have to sort of balance that with the potential for them to show up at the pumps. And you'll notice I haven't talked much about those, and I probably won't right now. That's a whole other thing. But really, and then this idea, and this comes from these action plans, of thinking about a terminal hatchery or some way to manage a stock-specific harvest. And I don't have the answers for that, but if we're going to have hatcheries, we're going to have to figure that out if we want to bring natural populations back. And then the question really is, can we become our own kind of salmon people with all of our modern culture and the sense of reciprocity? And again, that will be a challenge. And then a little exercise that we did in the group is on the left, we looked at a bunch of the existing initiatives, and this is in no way hyper quantitative. This is definitely back of the envelope. And on the right, we have a bunch of these needs that are sort of outlined to promote these habitat opportunities and also abundance, right, for that human salmon connection. And the top three initiatives at the left are initiatives that have authority. The other ones don't have authority. That's why they're got the dashed lines with their flow tubes there. And you'll notice that over on the right that, you know, there's not a lot of authority. There's some, but not a lot amongst all these strategies to really make these things happen. And in particular, the salmon cultural identity has a lot of great ideas, but there's nothing that's like that's really has like teeth. And that's really it. That's kind of where we're at. And now what we're doing is trying to think about examples of how you would take this framework and apply it. And again, not necessarily to critique existing strategies, but to think about how would you apply this idea, this holistic idea to managing salmon. And these are just some things like the idea of reintroduction, the idea of a winter pulse flow. And that's like the little snow globe, if anyone saw it. I like to show my little cartoon of that because it took me a long time to make it, but not today. And then the idea of fry releases, which are happening, they're going to be increasing. What's that going to mean? Is there a way to manage those to actually, I mean, to promote life history diversity as is in some of the plans as aspirations, but what does it mean? So I say thank you and challenge you to think about these things.
Oh, good question.
2 minutes.
Hey, thanks. I really appreciate your perspective and what you were sharing on this. Maybe I missed it earlier, but you were sharing these 3 guidelines. Where did these come from? Is this a working group or was there? Yeah, well, I think I may have missed that.
It's not a working group, actually. It's just a group of scientists that I interact with a lot, and we just kind of got together for a couple days, camping essentially, and just started generating these ideas. And it's sort of, you know, it's definitely the lowest thing on everyone's totem, whatever, I guess not totem pole, but it's lowest priority in everybody's list. So we're trying to get a little bit more energy and get out like a paper pretty soon. And I can go back to the co-author list if you want, but it's not really, we're not really representing necessarily agencies in this, even though we're all members of agencies so far. I blew everybody's minds. Carl, wait for the mic.
Good presentation. I think it's, you know, the way things need to be thought about, you know, from a holism perspective. But one thing I didn't see there was, you know, particularly rice fields and food supplementation, and how that might fit into those kinds of things.
Yeah, that's a really good point. And again, you know, I was rushing through this talk, and I think that kind of goes a little bit with the fry fish on on farm fields sort of thing. Yeah, I think that excuse me? Richard does incorporate that. Oh, the bridge group. Oh, there are groups that incorporate that, but they don't necessarily have authority right now. Yeah, it was on this. It was on the slide. I think Louise really put a lot of work into that slide, and so I apologize. I was a little stunned, but yeah, I think those are all. I mean, I'm not cutting anything out, but I don't think I really have time to talk at length about that because I'm out of time. But I'll talk to you about that on the side.
Thank you. Okay, so our next speaker is Andrea Chandler from Amélie Segarra's lab at UC Davis.
Hello, everyone. My name is Andrea Chandler, and I am a junior specialist in the SCAR lab at UC Davis. If you look closely in my first title slide, you can see a Chinook salmon there, which is a great segue to what I'll be talking about today, which is the detection of pathogens in ocean-caught Chinook salmon prior to freshwater entry. So I wanted to start off with just a super quick overview of the Chinook salmon life cycle. So salmon spawn in fresh water, then migrate to the ocean as juveniles, they mature, and then make their way back to freshwater as adults. And the life stage I'll be talking about today is those migrating adults. So the ones that are going back to freshwater in order to spawn. And during this migration, they undergo a multitude of migration stressors, such as poor water quality, high temperatures, predation, and pathogens. You might have guessed from the title of my presentation that today I'll be talking about pathogens. But what are pathogens? They are organisms that can cause disease. And this includes bacteria, viruses, and parasites. But since I'm talking about what can cause disease, I also need to mention the epidemiological triad. And this is a conceptual model that demonstrates how each one of our factors, our host, our pathogen, and our environment all interact to cause disease, because what each one of these factors alone could not cause disease. But why might we care about pathogens in terms of Chinook salmon? So pathogens can affect Chinook salmon populations in several ways. So first, infected individuals may be more sensitive to other environmental stressors. Also, California, including our San Francisco Bay Delta populations, are at the southernmost range of Chinook salmon. These populations are pushed towards their upper thermal limit, leaving them more susceptible to infection. In addition, warmer waters are in general correlated with higher instances of infection. So for our research goals, we want to monitor pathogens in adult Chinook salmon. So we want to know what pathogens are there, if any. And we want to know, is pathogen detection associated with adult migration success? Jumping right into our methods, we caught fall run Chinook salmon outside the San Francisco Bay Delta. If you look at the graph on your left, those orange circles are going to be our fishing locations. And we sampled for 2 consecutive years, 2022 and 2023. And I mention this because these were 2 very different water years. So 2022 had low amount of rainfall, there were warmer water temperatures, and in general, kind of poorer water quality. I believe there's some harmful algal blooms going on. While in 2023, it was a much wetter year, so there's a lot more rainfall and cooler water temperatures. So after the adult Chinook salmon were caught, several metrics were taken. So the ones you see here in that top picture is going to be energy density using a fat meter, and the picture on the bottom is length and weight. And I just want to take this opportunity during the slide to say that this study is part of a much larger project that aims to look at a bunch of different migration stressors. So it's much larger than the pathogen detection I'll be talking about today. But to simplify it, I'll just be talking about the relevant sampling methods. So as I mentioned, adult Chinook salmon were caught in the ocean. A gill biopsy was done, an acoustic tag was placed in the fish. Then the fish was released and the gill sample was stored. So at the end of the day, we had a sliver of gill sample and a corresponding unique acoustic tag ID. With that gill sample, we did our pathogen detection and quantification. So we quantified 47 of the most common pathogens known to infect salmon, and 47 is quite a lot. So in order to do this, we used Fludyme, a high throughput qPCR system, and you can see a picture of a Fludyme plate on your right. And we used Fludyme to detect and quantify the amount of pathogen DNA and RNA present in our gill samples. What this allows us to do is to assess, first of all, the presence and absence, and also the risk of infection. As you might have guessed, a higher quantity of pathogen DNA and RNA is associated with a higher risk of infection. All right, back to our research goals. So we specifically want to monitor pathogen detection in adult Chinook salmon prior to potential freshwater entry. We're going to do this by using qPCR to quantify the pathogens present in our gill samples. And to figure out its pathogen detection associated with adult migration success, we're going to combine that pathogen data with that telemetry data from our acoustic tags. So for our results, I just wanted to give you guys a quick visual of our pathogen quantification with this heat map. I don't want to spend too much time on it, but real quick, the x-axis is going to be our gill samples for both of our years, 2022 and 2023. And you can see we had 68 gill samples, so we sampled 68 fish in 2022, and we had 125 gill samples in 2023. And each one of those columns is going to be an individual fish. On our y-axis with all the text is going to be our 47 pathogens that we detected. And the colors are going to be a visual representation of the quantification that we got. So the yellow is low quantification, the orange is medium, and the red is high. And that dark blue is going to be the non-detects. So for pathogen prevalence, the bar graph on your left on the x-axis is going to be number of pathogens per fish. The y-axis is going to be prevalence. And I want to emphasize that we detected at least one pathogen in 91% of all Chinook salmon sampled, which was, I would say, more than we were expecting. We also found some co-detections. So for 34% of our samples, we had 2 pathogens detected, and for 16%, we had 3 pathogens detected. I also forgot to mention this on the last slide, but these stats are going to be for both years combined. So out of those 91% of samples, what pathogens did we detect? You can see the 16 pathogens that we detected on the x-axis here, and the y-axis is going to be prevalence of detection. So right away, you can see that CB/CIS is our most commonly detected pathogen, or Candidatus brachiomonas cysticola. So out of the samples that had a pathogen detection, 84% of those had CB/CIS, 33% had ichneal, otherwise known as white spot disease, and 12% had Flavobacterium psychrophyllum, which causes bacterial cold water disease. So an overview of CB/CIS. It's a bacterium. It's found in both fresh and salt water. And what happens with fish infected with CB/CIS is that cysts develop in the gill epithelia. And what can happen is that the gill lamellae fuse. So you can kind of see that in the picture to your left. And we think that we might have seen such a high quantification of CB/CIS in our samples because CB/CIS is multiplying and infecting the gills. So kind of makes sense. And infections can lead to respiratory distress, and in severe infections, it can cause mortality. So jumping to our locational data. So as a reminder of our methods, the fish were tagged and the gills were sampled in the ocean. And after the fish were tagged, as they moved throughout the Bay Delta, the tags were picked up by receivers put in specific locations around the Bay Delta. So we haven't finished running through all of the telemetry data, but what we do have is a list of fish IDs that were detected at Benicia. You can see Benicia labeled on my lovely map here. So this is useful because first, Benicia is a relatively narrow waterway compared to some of the other receiver sites that we've looked through, such as the Golden Gate Bridge. So this gives us more confidence in our detections. In addition, what we usually see is that fish that make it to Benicia are starting their freshwater migration. So they make it to Benicia and they continue their migration into freshwater. So what we're looking at for this, what we've considered for this study is that fish that make it to Benicia that are detected there have successfully started their freshwater migration. So this gives us 2 groups for analysis: fish that have made it to Benicia that have been detected there, and fish, for whatever reason, their tag was not detected by the Benicia receiver. And you can see in 2022, 27% of our tagged fish made it to Benicia, and in 2023, 68% of our tagged fish made it to Benicia. Okay, so tying together our pathogen data and our telemetry data. So we used a logistic regression model to assess whether pathogen concentration is associated with the odds of arriving at Benicia. We did this for all 16 of our detected pathogens, but CB/CIS was the only significant association between arrival at Benicia and pathogen quantification. And that's what you're seeing in this graph here to your left. The x-axis is going to be those 2 groups I talked about. So yes, arrival at Benicia, and no, did not arrive at Benicia. The y-axis is going to be our pathogen quantification units, which is log 10 copies of pathogen DNA and RNA per microliter. And what we found is that the group that was not detected at Benicia had a significantly higher pathogen quantification than the group that didn't. And another way to think about it or look at this is the odds ratio. So we looked at the odds ratio of the fish arriving at Benicia, and that is 0.7 or 70%. What this means is that for each one of our pathogen quantification units, again, that's log 10 copies per microliter of CB/CIS specifically, that fish had a 30% less chance of making it to Benicia. Okay, so, oh, I forgot my animation. There you go. So a summary of the results, 91% of all samples had at least one pathogen detection. The most commonly detected pathogen was CB/CIS by far, and a higher quantification of CB/CIS was associated with lower odds of making it to Benicia. In conclusion, is pathogen detection associated with adult migration success? So CB/CIS could be a potential factor in, again, adult Chinook salmon successfully starting their freshwater migration. But really, continued pathogen monitoring is needed to understand the potential impact of pathogens on adult Chinook salmon migration success. And I've also put the epidemiological triad here once again to remind you that this study is just a small piece in a very large and complicated puzzle. For future directions, we would like to include more locations from our telemetry data. So we have more receiver sites that go into fresh water, and we'd like to include those. And as I mentioned, the study is part of a much larger project to look at several factors, as I mentioned, like energy density. And we want to account for those other factors associated with migration success and to continue to build a model of what might be impacting Chinook salmon migration success. Thank you so much for listening. I'd like to thank the PI on this project, Dr. Miles Daniels, as well as my PI, Dr. Amelie Segarra, the Segarra Lab, the NOAA UCSC team, Ben, Cyril, Jeremy, Nate, and our funders. So funding was provided by the State Water Quality Control Board, and also Emmy, who is the other junior specialist in the Segarra Lab and who is doing a poster on pathogen detection in the McCloud River. So if you're interested, please check out her poster. Thank you very much.
We have some time for at least a couple questions.
Hi, sorry. You said that you detected pathogens, but did you see clinical evidence of illness in the fish?
Yes, absolutely. Great question. No, we did not. Since this was kind of more for monitoring as well, we didn't do any histology. We just look for pathogen detection. But yeah, that would be a great thing to add. It's just we're constrained and yeah, but that would be great to look at. Yeah.
Thank you, great talk. I was wondering if this pathogen in particular has any infection dynamics, or is it just it's caught in the environment by the salmon?
Yes, sorry, it's hard to locate people with the microphone. Yes, thank you. So it's hard to say because a lot of the studies that are done are in farmed salmon. So those are totally different dynamics than we're seeing in the wild. So basically, we're not sure. More study is needed. Definitely like some laboratory infections are needed to really know. And even then you can't account for all of the factors in the wild. Short answer, not sure.
Thank you. Very fascinating talk. I was wondering how you might expect the quantity of pathogens to change in various other areas of California, like I'm thinking more Northern California.
That is a great question. I will say there's not a lot of literature that looks at adult Chinook salmon pathogens. So even just like the monitoring is, I want to say relatively new, but someone please correct me if I'm wrong. So we don't even know what's really in our like Bay Delta system with the adults. So I am not sure what would happen with other areas. My only guess is that, as I mentioned, like higher temperatures are correlated with a risk of infection, but since so many other environmental factors kind of tie into it, it'd be hard to say, but it'd be really interesting to know. I'd love to know that.
I have one.
Yeah.
I saw that you have like very different rate of return at Benicia between the 2 years.
Yes.
So did you combine like both years when you did your logistic regression?
Yes, and that's kind of for more, because this is preliminary data, we looked at both years, but there actually are some yearly differences. I just didn't really we didn't have enough time to really look into it. But we did find some differences in 3 of the pathogens. So we did do that logistic regression on all of the detected pathogens between and compare the 2 years. We did find differences for 3 of them, and that's kind of also our next step to kind of compare those different years. Since, as you mentioned, we even had a different amount of fish that were caught and sampled, and part of that was because we think that the water quality was bad, so we just weren't able to catch a lot of like healthy fish, or and also by the 2nd year, the kind of whole project was a little more streamlined, so they were able to sample more gill samples in general. So that's also part of the higher number. But there is data like sampling for 2024, so I'd be really curious to see what that data is like for another year. Okay.
So you'd indicated a desire to look at other detection stations farther into the system. Do you have that data? And it would seem, there are lots of receivers out there. So, and is that going to be available, and are you going to be able to, or your project, be able to look at that and I guess get us a better idea of what the ultimate fates are?
Yes, definitely. And I will say I was not part of the like tagging team. So that would definitely be a great question for like the NOAA UCSC team. But yes, we do have information from the receivers. It just hasn't been like looked at and kind of processed yet. But yeah, exactly that. Since Benicia isn't even, it's brackish water. So as I said, it's like the start of freshwater, and it'd be really interesting to tie that into receivers actually located in freshwater.
Thank you on that.
Yeah.
Our next talk is a lightning talk, and it's from Cristina from the Southern Illinois University.
All right. Thanks for letting me back up onto the panel today. My colleague, Dr. Molinari, was not able to make it to this workshop, so I'll be presenting his data in his place. This project that I'm going to be talking about was in collaboration with Southern Illinois University, as well as the UC Davis group from the Cigar Lab. And we will be looking at the effects of dichlorodiphenyl dichloroethylene, or DDE, on Chinook salmon outmigration success. So throughout this study, we saw that there was a concern regarding juvenile Chinook salmon exposures to DDE, and that multiple behavioral endpoints were impacted. So if you guys know what DDE is, you will likely know that DDE is found everywhere. It is one of the most consistently detected compounds in California, including just the watersheds, the sediments, the waters itself, as well as the terrestrial lands. But this is not just a problem associated with California. This problem is found globally. And so DDE concentrations are detected everywhere from US to China to Africa, and all of these different exposures are associated with different adverse effects. A little bit of background on what DDE is. It is one of the main degradants of the product DDT, or diphenyl trichloroethane. It was popularized for its use in World War II and banned in the 1970s from the EPA based off of different effects that they saw on bird populations. But this was not only something that impacted birds, but also aquatic life. And usually the way that you saw concentrations for these effects were looking at external concentrations like waters, sediments, and the diets. But when you look at these different external factors, you have to take into consideration things of bioavailability or the routes of ingestion to see how it gets into the fish itself. So more so of an association rather than a direct measurement, unlike when you look at internal body residues, where you take the tissues of the fish, and those different factors are not taken into consideration because you're measuring the direct concentration that's associated with the fish tissues itself, especially for something that is hydrophobic like DDE, where they'll bind to the lipids or the organic matter of the sites. And so using this internal body residues approach, we were going to be measuring a couple of different exposures of DDE to salmon during their juvenile stages to see whether or not that these sublethal concentrations of DDE impacted any of their outmigration behaviors. And so the behaviors that we're going to be assessing today is swim performance, growth, photomotor response, routine behaviors, shoaling behavior, and anxiety-like behavior. The first two were done at SIU, and the second two were done at UC Davis. Out of these 6 different parameters, 3 of them were significantly different or significantly impacted by DDE exposure, but we're going to go for the first 2 first. So there was no impact on a DDE residue for both swim performance or growth for Chinook salmon. For their swim performance, you have the lipid normalized DDE concentrations on the bottom, and on the y-axis, you have their max bursting speed. And regardless of going from environmentally relevant concentrations of DDE inside of for the exposures to 100-fold the concentrations of environmentally relevant, there was no impact on that swim performance. For the growth rates, we have, again, the lipid normalized DDE residues on the x-axis and the growth rate for length or weight on the y-axis. And again, just like swim performance, there was no significant impact with those DDE exposures to their growth, as also seen by their low R-squared values for length and weight. For the anxiety-like behaviors, we have that on the upper left corner up over here, where we have the treatments on the x-axis and the portion of time spent in for the anxiety was measured by thingmataxis, which is essentially how long the fish will spend around the borders of a tank versus the center. And regardless of the treatment, they would all spend the majority of time towards the border of the tanks that they're in for, so there was no significant impact. For shoaling behavior, there was an increase in each of the treatments that we're seeing compared to their control. And so for the 14 to 953 nanomoles per gram lipid, there was that increase in the proximity the fish would spend next to each other. For routine behavior, there was a variety of significances where there was a decrease in the distance moved and velocity for the highest treatments. There was a decrease in freezing for the middle treatment. There was an increase in bursting behavior for the lowest, and for cruising behavior, there was an increase in both the median and the high treatment. And lastly, for photomotor response, which is essentially the response to light stimulus and the freezing that they would experience afterwards, there was a significant increase in that freezing time for the lowest and the highest treatments. So bunch of different treatments towards the fish, a lot of different results, but ultimately what this is essentially showing is that the outmigration behaviors are impacted. Any changes that happen in regarding to routine behavior for the exposures will showcase that there is a possible increase of predation for the juvenile Chinook salmons. Anything for the increase in the proximity could show or indicate that the salmon are under any sort of stress, but also it'll increase the amount of competition between the fish for itself for other resources like food. And so all of these different impacts are essentially impacting their success of outmigration. Keep in mind that the study was done under a 10-day period, and so any other effects more for longer chronic exposures, such as endocrine disruption or intersexuality, which is associated with compounds like DD, may have went undetected during this testing period, as well as any other kind of narcotic effects from that contaminant exposure. Right now, we're currently working on other studies looking at other consistently detected compounds in the San Francisco estuary or the California Delta, including fipronil and bifenthrin, which is a current use pyrethroid insecticide found in a lot of the sites, as well as mixtures, since these contaminants don't exist alone. Yeah, so thanks again for our partners at UC Davis, as well as the California Department of Fish and Wildlife for funding this project. If you guys have any questions, let me know.
I think we have the time for questions. So if you have any questions, please feel free to reach out to her directly. Thank you. Our next speaker is Luis Cominasi from the Segara Lab at UC Davis.
Hello, thank you for the introduction, and thank you for Cristina, because this study is part of the same project, so hopefully it's not too repetitive. But I'm gonna start with a bit of a background of why we're doing that. So California has the greatest concentration of at-risk salmon, with 31 species that are all in decline, and Chinook salmon is no exception. The population has been declining since the 1800s, and for the past decade, this decline is even more profound, which has conducted to list the winter wren and the spring wren as endangered and threatened. So why is the population declining? So the factor are multiple and complex, but it can kind of be boiled down to habitat alteration and how it changed over the years. So in the past, it was mostly freshwater and seasonal wetland, and now it's occupied by agriculture and urban area. And with this increase in urban and agriculture, came the increase of use of contaminant and insecticide, and that running off in different water body during storm events. In addition to that, the insecticide are predicted to increase, or the use of insecticide are predicted to increase by 4 to 170-fold by 2099. So a study by USGS and the Delta Regional Program Monitoring have observed in 2017 that at different locations in the Delta, between 5 to 20 different contaminants were detected at a time, and listed about 154 different contaminants. And among them, commonly detected insecticide were bifenthrin, fipronil, and DD. And as Cristina mentioned, DD is a byproduct of DDT, which had been banned in 1972, but is still detected to this day in sediment and the water column. So those are used for pest and insect control and act as a neurotoxic on the insect, which leads to death. But unfortunately, it also contaminate non-targeted species like fish in the water. And it's often hypothesized that stressor together their effect will be additive, but they can also be smaller than the effect of one stressor or beyond the addition of the two. So our research question here and what we were interested in looking at is the impact of this mixture of insecticide on juvenile Chinook behavior, and not only with water exposure, but also bioaccumulation that happen via the food chain. So for that, we had different treatment of exposure. So we had our juvenile that were 4 to 5 years old that were exposed to either a control, a low treatment, so with low concentration of bifenthrin and fipronil, a high treatment with high concentration of bifenthrin and fipronil, and a tertiary mixture with bifenthrin, fipronil at low concentration, plus some DDE. And those concentrations have been chosen to reflect after the 9-day the body residue that we have seen in wild-caught fish. And so what we did, we transfer the fish in an arena, they acclimate in the dark, and then we record the behavior in the light and observe different parameters of locomotion, such as total distance move, velocity, cruising, bursting, or freezing. We also look into the anxiety-related behavior called chemotaxis or wall hugging. So naturally, the fish will tend to stay at the border of the arena, but if they're less anxious, they tend to be more bold and explore the center of the arena. And then we looked into the social interaction of the fish or shoaling. So fish use shoaling as a strategy often against predator with dilution or confusion of the predators. So now for the results, starting with locomotion. So each of the graph is one parameter of locomotion, and our different treatment are always compared to the control. So if we start by looking at the low concentration of a binary mixture of fipronil and bifenthrin, we can see that there's no effect in freezing, bursting, or cruising, but there's a slight increase in distance move or velocity. And our same binary exposure, but with higher concentration, here we see a different pattern. The fish are less active, they freeze more, and they're not as fast. So we talk about hypoactivity. And again, with the tertiary mixture, we see the same effect. So we actually have the low concentration of fipronil and bifenthrin, but with the DDD, the effect is closer to high concentration. So if we compare with previous study that been done on single exposure, there were no effect of single insecticide exposure of fipronil and DDD, but an effect of bifenthrin that lead to hypoactivity. But for a mixture at low, there were slight increase in activity, so it doesn't reflect any of the effect of single exposure. And for the high and low plus DDD, we are seeing hypoactivity, which reflects what we saw with only bifenthrin. Now, if you look at chemotaxis or wall hugging, so here for every treatment, the chemotaxis or like anxiety level actually increased. They were not exploring the arena as much. In the graph, sorry, I didn't explain, but the lighter orange mean that they're close, like often at the border, and darker mean that they're close, exploring the center. And again, if we compare with single insecticide exposure with only bifenthrin and fipronil, they were less anxious, so more bold, and no effect were observed for DD. But now with the mixture, we are seeing the opposite effect. They are more anxious and stay really hidden. And if we look at our last result about shoaling behavior, here we're looking at the distance between fish. And the distance between our fish is actually increasing with the mixture, the concentration, but also the tertiary mixture. We can see a more profound increase. So I put a down arrow because, I mean, the distance increased, but this shoaling behavior is decreasing. They are not staying as a shoal. And I think it was a surprising effect because at single exposure, bifenthrin, the shoaling behavior was decreased, fipronil too, but DD, they tend to get closer together. So we thought it was gonna counteract the effect of bifenthrin, fipronil, but actually the mixture of the three, we see a more profound effect and the disruption of this shoal. So here, basically, as we increase the concentration of the insecticide and we multiply the number of stressor, the shoaling behavior decrease greater. So to kind of summarize everything, we saw that mixture of bifenthrin, fipronil, and DD had a significantly altered behavior of juvenile Chinook, and it was also specific to the different endpoints. We saw an hypoactivity, an increase of the anxiety-related behavior, and a reduction in shoaling. And it's going to have impact on foraging. For example, if they're hypoactive, if they're more anxious, they might hide more and don't go out, like not hiding. They are hiding, so they don't go for food. And if they're hypoactive, they might just not be as fast or freeze more. So it might be dangerous when they are around predators. And same for the reduction in shoaling. I established that shoaling is a good strategy against predators. So if they're not shoaling as much, they might be more subjected to predator as well. And probably other impact, we don't know what it means, the hyperactivity, if they are they out-migrate and they encounter different flow. So all of those pesticides and this mixture just add pressure to this population that is declining. And for future direction, so this study kind of highlight how it's important to not look only at one pesticide, but the mixture of the pesticide together, specifically with different endpoints. They are endpoint specific. And we also are currently doing another study that assess bioaccumulation in fish that we deploy in the Delta. So we started last year and we're repeating it this year, so in a few months. But the only thing that we haven't done and need to be done in the future is deploying fish and then measuring their behavior in the same kind of endpoint after with relevant environmental condition. And so I would like to thank the Sager Lab, but also the Lighting Lab that worked on this project and our funding. Thank you.
Hi there. I noticed that that the variability in behavior seemed to go up with more with higher intensity treatments, and I'm just curious why.
So it's the pathway of, for example, bifenthrin. It's kind of to excite the nervous system and the passage of different ion. So I'm not exactly sure of the mechanism behind it, but it seems like depending on how much the channel that the ion pass, it affect the brain, that they will have like be more or less active. So there's definitely a change happening at the neuronal level that we're not sure exactly what's happening, and I haven't measured, so I cannot tell you, but it's something related to that.
Thank you.
I have one. The concentrations for your low and high treatments of the bifenthrin and fipronil, is that low and high treatment reasonable for what is measured in the field?
Yeah, the low is definitely what we find in the environment. The high is a little bit higher than is usually found. So it would be maybe in systems that are kind of more close and not as have less flow. But yeah, the lowest is definitely something we see and measure and detect, but the high would be like
it's possible, the high.
It's possible, maybe like I mentioned with climate change and the use of more insecticide, it's coming, basically.
Thanks.
Any other question? Yeah, sure.
I have another one. How did you choose to include 3 fish in your trials versus 5 or 6? Because that might affect the behavior or dynamics of the shoaling behaviors.
Yeah, I just did literature. I haven't tested, but some papers showed that you could see the same behavior in 3 fish than 5. So I just trusted them.
Thank you, Luis. Our next speaker is Pete Wynn from Bruce Amok's lab at UC Davis.
Hello, everyone. I'm a little nervous, so be kind. All right, so today I wanted to talk about delta smelt during the lava stage. So according to a Bennett paper in 2005, the famous paper, he estimated that like delta smelt during the early stage have a really sensitive early life stage, and with like only 1 to 5% of eggs were able to grow and mature to become an adult. And even when you look at delta smelt in an environment that lack predation pressure, like a fish hatchery at FCCL, like their survival of larvae is still only around 47%, 40 days into their larval production. And in my opinion, our understanding for what is the optimal condition for delta smelt larvae and what environmental factor that drive these species to be so much successful in the past, like we still don't have like a clear answer for them. And here's a photo of delta smelt larvae. They are a tiny translucent fish with only around like 5 millimeters in length. They can able to swim almost right away and start eating like around 3 to 5 days after they hatch. Their main diet consists of zoo small plankton ranging from copepod napoli to mysis as they continue to grow. And unfortunately for delta smelt, zooplankton production has been declined since the 1990s due to the suppression of the phytoplankton caused by several anthropogenic factors. And this could be very important to smelt just because in general, they have a relatively small yolk sac. So as soon as they absorbing all that energy that they store there, they failing to capture prey could lead them to starvation, and in other words, food limitation stress. Besides the decline of the zooplankton, salinity variability is also another factor that had changed in the estuary, so much that the salinity in the estuary has remained almost kind of the same. Instead of experience like a fluctuation of salinity across regions, season, or depends on the water year. And as we all know, salinity are one of the key factors that affect fish species, survival, metabolism, and distribution. And for delta smelt larvae, this could be a very important. Again, they are really tiny. They have a really high surface area to volume ratio. They often lack osmotic regulation organs such as gill, which make them very susceptible to osmotic stress and affect their growth and survival. All right, so today, in general, I have two objectives. I wanted to use observational data to kind of describe what is the current health condition, diet, and distribution of delta smelt larvae, and also using experimental data to kind of summarize the effect of summaries on their survival and growth. All right, let's talk about the deep water ship channel. According to the recent field data, this might be the only location in the estuary that still have a relatively high abundance of larvae that still exists. Due to its structure, this man-made 43 miles long channel would act like a dead-end slough. And because of that, they create a gradient of water exchange throughout the deep water ship channel. And as you can see in the lower region of the deep water ship channel, there will be a really high level of water exchange. And as you move upstream, you will see a decline in the level of the exchange. And when you go into the upper region, there will be no water exchange at all. And this create a layer of complexity within this location and make these regions very uniquely different in terms of water quality. For example, the upper region of the deep water ship channel the water would be really clear, where the middle region would have a really high turbidity, and zooplankton would be very abundant in the upper region, while as they move downstream, zooplankton is just not there. So by looking at how the Smelt larvae respond throughout these different water quality gradient, we can kind of make some sort of connection between what environmental factors are affecting their health condition. All right, so to do that, we use the larvae that were collected from the EDSM 20 millimeter survey from April to June from 2019 to 2022. We use catch per unit effort, CPUE, to discuss their distribution. Half of them get processed for histology to kind of in general look at their health condition, and the gut content, half of them will get processed for gut content analysis done by the CDFW to look at their diet and stomach fullness. All right, so for their distribution, if you look at the y-axis here, if you look at the average CPUE on the y-axis, you can see that delta Smelt or larvae are most abundant in 2019, the wet year of 2019, and the middle region of the deep water ship channel. And this is a very interesting data just because the upper region have a really high zooplankton abundance, but we don't see larvae there. And since the middle region are associated with turbidity, we ran a separate analysis for turbidity for CPUE, and we found that turbidity have a significant effects on CPUE, which peak at 30 to 40 NTU. But when turbidity is lower than 20, we barely see any larvae. All right, in terms of their health condition, the average gut fullness is around 42%. Within this data, 20% have an empty gut, and I was at 12% have an empty gut. That's my mistake. And 57% have a 20% full gut. No larvae have any gut content at more than 75%. When you look at their histology, you can see that 50% of liver have a severe glycogen depletion, and 26% have a liver lesion score to be 3 or more. And if you look at unhealthy liver, you can see like a clear definition of the liver cell, and the little white speck indicated by the black arrow for glycogen. But if you look at the unhealthy liver, you can see that the cells start to fuse together with a darker nuclei, indication of liver cell breaking down itself due to the lack of nutrition. And for reference, the very similar lesion score and lesion type were found in subadult that have been fasting for 1 month at 16 degrees Celsius. No significant difference that we found in histology in terms across region or year. And we didn't see any lesion in the gill as well, like the gill is in excellent condition. In terms of their diet, Cynocalinus species are the dominant prey item for them in both number and prey mass, and followed by the uri and basuto. This shift in their diet, the effect of them on larvae is still pretty unknown, just because they all have different type of nutritional profile, and further experiment need to be done to kind of confirm that. I also wanted to point out is that as larvae continue to grow, they tend to have a more diverse diet in their gut. And this emphasizes how important growth is to larvae. All right, let's talk about some experimental data. To test the salinity on the effect of salinity on larvae, we basically expose newly hatched larvae to a range of salinity from freshwater to 18 ppt. We have total of 9 treatment in the middle and 2 replicate each. We expose them for 45 days at 12 degrees Celsius. We collect larvae throughout the experiment to track for growth and count every survival larvae at the end for final survival. All right, if you look at growth, you can see that growth increase as salinity increase, and growth does seem to do best at 8 ppt. In terms of survival, very similar pattern we see, like the higher salinity, the more survival of larvae, with peak around 4 to 8. We saw a lot of mortality in 18 ppt treatment, but this is like an exceptional just because we ran the experiment for so long and keep adding algae into the bucket. It get overgrown. Algae got like a little algae scarp around them. They probably die because of that. Well, one thing that I want to mention is that like the food item that we use for these experiment are rotifer and atemia. And these guys are sea-like creature, so they like higher salinity treatment, so they tend to be more available in the water column, allow larvae there to kind of eat more. So that's a potential co-founding factors that we should consider when we make any kind of conclusion. All right, a second question that I ask is there any interaction between salinity and temperature at all? All right, so to test this, we expose newly hatched larvae to a range of temperatures from 13 degrees Celsius to 21 degrees Celsius under 2 salinity condition, 4 ppt, and fresh water. I use one recirculate system each for each salinity, and one system would hold 5 temperature, the other one would hold 3, with 3 replicate each in one, 4 replicate in the other. All right, feeding. Unfortunately, I fed these guys very differently, and my excuse is that my rotifer culture just kind of crashed right before I started the first trial. And since the survival in the first trial is not that good, spoiler, I decided to just feed them as much as possible in the second trial. We ran them for 14 days, and here and here's the beautiful recirculate system that I built. I'm not going to go over it. I'm just not saying it's good. But then all temperature treatment basically just share the same water quality. So to remove any type of co-founding factor in terms of water quality. All right, so for survival, all right, if you look at these little blue dots represent final survival for each replicate in the freshwater treatment, you can see a dramatic decline in survival within the low feeding condition. But when you, as temperature increase, right? But then as you increase the feeding, larvae tend to do a little bit better, but still displays a very similar pattern. For brackish water, however, those red dots just kind of like stay in the same place across temperatures. And if you feed the fish more, they really like them in brackish water. And we also lost some replicate during especially in the second trial when we feed them a little bit, like way too much. So that's my fault, I guess. All right, in terms of length, all right, this is a very interesting graph because under low feeding, you can still see that larvae grow better as temperature increase, but then you see a small dip at 21 degrees Celsius. Unfortunately, for the second trial, we didn't run any temperatures that higher than that, but you can see that the larvae outperforming within the low feeding. For freshwater treatment, you see very similar pattern within the high feeding treatment, but then for the low feeding treatment, you see almost no growth at all within the freshwater treatment. Very similar pattern you see in weight is almost identical graph. So like, I don't want to talk about it. All right. in summary, I know there's a lot. All right, so based on the gut fullness data and the histology data, we found like evidence of food limitation stress within the dead smelt larvae in the deep watership channel. And since we only look at the larvae here at this location, I can't tell if the other location in the estuary or the larvae are experiencing the same thing. Also, turbidity is another factor, a key factor more than zooplankton. Have shown that affecting the distribution of larvae population in the deep watership channel. And I, in general, would recommend some sort of management action to be target habitat to be more than 20 NTU, just because that's where larvae live. We show that using experimental data, brackish water have the positive effect on growth and survival. And feeding, like even though my study doesn't design to test feeding, we kind of see a glimpse of how important feeding is to these guys. And especially when they're in fresh water. All right, final message is that I want to highlight how sensitive fish species is at the larva stage. And I know that they're really hard to work with as someone that work with them every year. Like, but they deserve our attention because it doesn't make them any less important to the estuary. I want to say thanks to everybody at the FCCL for providing the embryo, the Aquatic Health Program, Bruce Hammock specifically for provide all the help, the U.S. Fish and Wildlife for all the beautiful survey that they do, the CDFW, I never met these you guys, so please come meet me. I don't know. And thank you for the Bureau of Reclamation for all the funding for all this beautiful data. And questions? Yeah.
So the middle section of the deep water shipping channel had the turbidities that you found that the larval fish liked. Were the salinities there in line with what you found in the laboratory were conducive to their happiness and health?
I think like in general, like the salinity was kind of higher actually in the upper region since there's no water exchange. Like we didn't really take a look at these data because like the salinity is important, but then for some reason like they just not important in this case. And the increase in salinity is like very slightly, like it's not like you go from like completely fresh to like really high. So like we could potentially test that, but and again, like the first part of the paper is from someone else. I'm just kind of like present their data in a way, so I don't really have a say to it. But yeah, like I would love to look at the interaction between salinity and larvae there. Yeah.
We have a question from Dennis Cockrell. Do you think the brackish water is better for the zooplankton, like as a feeding source, equaling better conditioned plankton source in the tank versus water conditions being more optimal for fish itself?
I think in general, like the phytoplankton level in the deep water ship channel is tend to be higher compared to other area. Correct me if I'm wrong because I'm not like a phytoplankton or zooplankton person. But I think that like zooplankton could be important there, and especially for in terms of hatchery. Like one thing that we like that, like I don't want to criticize FCCL, but they feeding they feeding a freshwater larvae using a sea-like creature like rotifer and atemia, and of course it potentially affect clearly affect their survival within the hatchery. But then again, let's talk about like how not how bad we are at keeping freshwater larvae in general, because the field of marine larvae has been so much better compared to freshwater larvae. Nobody really care about them, basically. So yeah, that's my answer. I don't know if yeah.
We have time for another question here, right?
Hi, good talk. Enjoyed it. I had a question about the gut content analysis. I know you listed Pseudodeoptimus, Temra, Cynocalamus. I assume that's done with molecular methods?
Yeah,
It's okay. I'll continue my question. Sorry. I was just curious if anyone compared the molecular results with visual analysis to determine whether it was copepod eggs or napuli, or if it's just an assumption that digested prey is mostly napuli and not eggs.
Yeah, I think the CDFW lab, they just look at them like I just identify them like very visually. I don't think they do any sort of molecular DNA thing. I know that like we did do a DNA analysis for each larvae to just kind of make sure that they are Delta Smelt, and that's like the only thing that I know about like if they use DNA or not. Yeah.
Okay, thank you.
Great question. So from my understanding,
they're just
so you found no significant difference for region or year for lesions and glycogen stores. I found that pretty interesting because it did look like you saw regional differences for zooplankton, turbidity, and chlorophyll. A lot of our work is based on the premise that if we provide food, the fish will eat it.
Yeah.
They didn't seem to eat it, and I wonder if you have a suspicion for why.
Again, like this is just a snapshot of how these fish larvae are out there, right? So for me, you know, a lot of things could depends on like is the time of the survey a little bit earlier. We don't, and again, we don't really know exactly when they feed, and do they feed at night? I don't know. Like so like again, this is just like a snapshot of the gut foodness, and could potentially, if you survey them a little bit more earlier, you may see the gut more full, or a lot later. And again, that's why like the histology data is there to just kind of like assess. I think like the gut fullness data is mainly for like feeding success in general, and yeah, like that's. Yeah, I don't really have like a clear answer for that. But in general, their condition just kind of bad overall all the time.
We are still on time with our schedule, so I will use that time to ask one question. So I saw that you have a higher growth with the higher salinity. So do you think that the delta smelt larvae have a higher aerobic scope at this higher salinity that could explain this higher capacity for growth?
So I think like in general, like the delta smelt larvae have a really high tolerance for temperatures, and as they grow older, they become more sensitive to temperatures. But again, like I think a lot of their growth is if they require like a lot of energy just so that they can grow and get out of that size where they're really sensitive to anything. So like that's why like and you can kind of see that like the growth that you saw, like I think like according to like Levi paper, you can see that the growth that you see for survival larvae within the for survival adult, the growth that they have in the larva stage are way much more compared to what we see in the hatchery. So in general, they require a lot more energy than we think they do to kind of successfully grow and survive. I don't know if that answered your question.
Thank you. Our next talk is actually a lightning talk by Jackie Lang from the Sagara lab at UC Davis.
Yes, hi, I'm Jackie Lang, and today I'm very quickly going to be telling you about this part of my project, which is a small part of a much larger project that I've been doing for my PhD. Let's see. There we go. Okay, so about 30 years ago, people up near Seattle were observing coho salmon, adult coho salmon that looked similar to the one in this photo on the left. And the symptoms that they were seeing are that they would be surface swimming and losing their equilibrium, and they had increased gasping and ventilation. And when they started seeing these, they were observing these symptomatic coho, and within a few hours, almost all of them would experience mortality. And only 4 years ago, researchers up in Seattle figured out that a chemical that is associated with tire rubber called 6-ppd-quinone was causing this mortality. And so they first figured out the LC50, which stands for lethal concentration for 50% of the population. And they found it at 0.095 micrograms per liter, which is extremely low, meaning that it's very, very toxic to these coho salmon. And these researchers also measured different environmental samples. And if you look at the far left part of this figure on the right, the far right section of the figure on the right in yellow, we see that concentrations that have been measured in the San Francisco Bay Area are kind of hovering right around that LC50 and sometimes even exceed it. So our question is, how is 6-ppd-quinone affecting our endangered smelt? So we're looking at both longfin smelt and delta smelt, and we've tested it in both larvae and juveniles. And so we looked at a concentration range from 0.1 to 100 micrograms per liter. I just want to highlight that the 100 micrograms per liter is many orders of magnitude higher than concentrations that we see in the environment. But luckily for our smelt, we saw no mortality, which was kind of surprising because, like Kit just mentioned, these are very fragile fish and you can just look at them wrong and they go belly up, but they were fine this time. So our next question was, are there any sublethal effects that we might have to worry about with these fish? So first we looked at effects to behavior, and to do that, we exposed fish to sublethal concentrations for 96 hours and then recorded their swimming behavior over the course of 30 minutes. This is a lot of information, and I'm still kind of analyzing the data, so I'm not going to get into the weeds of it, but basically what we're seeing is that for both longfin smelt and delta smelt, there are behavioral changes due to 6-ppd-quinone exposure, and even at environmentally relevant concentrations. And often, but not always, when we see behavior changes due to contaminant exposure, it suggests that there might be a neurotoxic element going on. We still don't know exactly what the mechanism of toxicity is for 6-ppd-quinone, but this does kind of lend to a growing body of evidence that 6-ppd-quinone may be acting as a neurotoxicant. And then using that same experimental setup, we were also able to measure changes in thigmotaxis, which is anxiety-like behavior. And to do that, we assess the amount of time that the larvae spend wall hugging, which kind of suggests they might be feeling a little anxious compared to swimming throughout the center of their enclosure, which might suggest more bold behavior. And again, I'm still analyzing this data, but for both species, we are seeing that at environmentally relevant concentrations, we're seeing changes in thigmotaxis due to 6-ppd-quinone exposure. And changes in thigmotaxis and also behavior can impact these larvae's ability to perform essential biological functions, including capturing prey and escaping predation. And so this may be affecting individual survival and overall population health as well. We were also interested in looking at potential cardiorespiratory effects, mostly because of those observations of increased gasping and ventilation in the field. And so to do that in our larvae, we exposed them to sublethal concentrations and then anesthetized them under a microscope. And we slowly ramp up the temperature and measure their ability to increase their heart rate to cope with that thermal stress. So on the left here is kind of just a graph showing what that tends to look like. We tend to see an increase in heart rate And then at a certain point, it kind of starts dropping. They aren't really doing as well. And then eventually we see arrhythmia, which is an irregular heartbeat. So on the right here, on the y-axis, we have the temperature that they're experiencing that arrhythmia, and then on the x, the concentration that they were exposed to. And in Longfin smelt, we saw no differences compared to the control, but in Delta smelt exposed to very high concentrations of 6-ppd quinone, they actually seem to do a little bit better than the controls. And so what we think is happening there is that during their sublethal exposure, they were feeling stressed and potentially upregulating some of their stress response mechanisms. And so once they were exposed to that thermal stress, they were kind of already primed to be able to accommodate for that compared to the controls. And so in conclusion, we did not see any mortality, even at really, really high concentrations in both larval and juvenile longfin and delta smelt. And then 6-ppd quinone induces changes to behavior and thigmotaxis in larval longfin and delta smelt. And 6-ppd quinone doesn't increase thermal tolerance in larval longfin or delta smelt, at least at environmentally relevant concentrations. But overall, I want to highlight that the results of this presentation and also some of the others that we've seen in this session show that if we only are focusing on lethality, we may be missing relevant sublethal effects and underestimating risk to these vulnerable species. So I think I have a little bit of time for questions. Oh. And thank you to my lab and funding agency.
Question? I won't ask one because I'm introductor on this presentation. Okay, thank you, Jackie.
Great, thanks.
And so our last speaker is Jamie Byrne from Kramer Fish Science.
Hi, everyone. Thanks for hanging around today. My name's Jamie. I am a technician with Kramer Fish Sciences. And for the past 2 years, we've been studying the survival of Chinook salmon eggs and the associated hyperate conditions during incubation in the Sacramento River. So winter run Chinook salmon are the only population of Chinook known to have a true winter run life history, and they are known exclusively to the Sacramento River. Historically, they spawned in the cold water tributaries of the Sac, shown in black of this map. And then after the construction of the Keswick and Shasta dams, their spawning habitat was greatly reduced, and they're now restricted to the warmer water temperatures below Keswick. And as we all know, this greatly reduced their population and eventually led them to being listed as an endangered species. And this called into question their success of incubation. So while there's some efforts being placed to kind of mitigate this, such as cold water releases from Shasta Dam, there overall is limited data known about the survival of eggs and the associated conditions within the hypereic zone. So that's kind of the overall goal of our study, is to gather more data on this topic and more specifically investigate the impacts of substrate and temperature. So for our study, we picked our sites based off of known spawning locations of winter run Chinook salmon, as well as sites with suitable substrate and depth and velocities. For our first year of our study in 2023, we had sites B, C, and D. B was our furthest upstream near Turtle Bay. C was our middle near Bonnie View Boat Ramp, and site D was our furthest downstream beneath the confluence of Cow Creek. And then for our 2024 year, we added a deep water site just below Keswick Dam, which is our site A. And this site is characterized by a gravel injection pit, which is shown on the bottom picture on the left. And what this does is it's deposits substrate of clean, suitable size for spawning into the river to help create more spawning habitat. And since this location had a depth of about 15 feet, we used divers to help us here. So in order to do this, we dug 10 artificial redds at each of our sites. And here's an example of one with the arrow pointing to the pot. And then we retrieved fertilized fall run eggs from Coleman Fish Hatchery. And although this study was focused on winter run, we used fall run as our surrogates to minimize our impact on an already struggling species. Then we took our fertilized eggs and put them in these modified incubation boxes along with temperature loggers. And we buried them into our artificial redds and let them incubate in the river for about 2 months from October to December. Then using a temperature-based model, we predicted about 50% hatch plus some extra time, and then we went back and retrieved them. And we separated out our hatched alvin and sediment less than 2 millimeters, and we took these back to the lab for processing. So what we found for our 2023 year was an overall trend of survival decreasing, moving from upstream to downstream. And then in 2024, with our addition of our deep water site, we found that our trend was consistent with our proportion of eggs survived to hatch was highest upstream at our site A and steadily decreased with the least surviving at our downstream site, site D. And for our average mean daily red temperatures, we found a trend of lowest overall temperatures at our furthest upstream site with the least amount of variation. And then moving downstream, we saw higher temperatures with more variation. And we saw that overall across all sites that the temperature did not exceed our known mortality threshold of 13 degrees Celsius. And these trends were consistent across both years of our study, with our 2024 year seeing just slightly overall temperatures. So when comparing our temperature data to some flow data, we see that during a significant storm event that occurred around Julian Day 325, which is about late November, at our upstream flow gauge by Keswick Dam, there was a small flow increase of less than 1,000 CFS. Meanwhile, at our furthest downstream flow gauge near Bend Bridge, there was a flow increase of over 40,000 CFS. And the y-axis on the flow graph is not to scale. But this led us to believe that our site D temperatures and flows were being heavily influenced by weather events. In regards to our fines, we saw, again, a trend of fine sediment less than 0.85 millimeters, increasing from upstream to downstream. We saw the least amount of accumulation of 0.85 fines at our site A, and then this increased as we moved downstream to site D. And we saw that 0.85 fines having effect on our survival at our sites A, B, and D. So overall, we saw that temperature fines and survival all co-vary. However, since our temperatures never exceeded our mortality threshold, that leads us to believe that fines is our driving mechanism for survival. However, our indicator mechanism is unknown. Some of the things that we are speculating is that the fines are coating the eggs, leading to suffocation, or clogging the interstitial space, which is decreasing permeability. Or it could possibly be contaminants coming from stream runoff. However, temperature and fines do strongly co-vary, so these results are a little hard to tease apart. Currently in our lab, we are working on processing our alvin samples to quantify development and health property metrics. And we are also hoping to follow up with a sort of field controlled study to look into our fines and survival a little bit more. And I'd like to thank Sacramento River Settlement Contractors for funding this project. And all of the staff at Kramer Fish Sciences, especially my mentor, Jasmine Williamson, and project manager, Steve Zoig, as well as Big Valley Divers for helping us at our deep water site.
Any questions?
Thank you. What is driving the fines other than just location? Are they dropping out lower in the main stem, or do you think there is like a tributary coming in there that had a wildfire? Do you have a sense of where that's coming from?
Yeah, so the most amount of fines we saw was at our site D, and site D was right below the confluence of Cow Creek. So we think that's like the main driving factor of why we saw so much fines there, especially after a big rain event that Cow Creek became really inundated, and we saw a lot of flow coming out from there. So we think that's probably one of the big factors in it.
Any other question?
Good talk. Just out of curiosity, I was wondering if you guys ran any like principal component analysis on the different variables on that, just to see whether or not there was any significance.
I'm sorry, one more time.
I was wondering for the data based off of the just like hatching and whatnot, if you ran any principal component analysis. It's a non-parametric statistical test to see whether or not from those numeric values, if there is a significant correlation between those different parameters.
Not entirely sure.
Yeah, we have a question online. How confident are you that the cold water temperature threshold is actually the right number? And shouldn't it be a degree or two lower?
So not entirely confident. That temperature threshold is based off of some lab studies that have been done. But that threshold has been a topic of discussion. But we have just used it as our threshold for this study. But it is something that is being looked into and followed up on.
Thank you, Jamie. So that's the end of our session, and now I think we can all go to the poster session.
All right, thanks everybody, all our presenters for an excellent day two. As he said, we will be doing the poster session until 5:30. Take all your belongings with you because this room will be locked up soon. If you're not coming back tomorrow, please be sure to leave your badge holder at the registration table so we can use it again, and we will see you tomorrow. Thanks.